So once again, today's event is being recorded. We'll be providing the recordings of the presentations in this session only, not in the breakout rooms. After the event, we'll post those on our website. And we actually will still have people walking around taking photos throughout the day, just of general conference attendees, but no videographer today. Once again, restrooms are, if you go out to the main lobby area, past the elevators to the left, the restrooms are right there. And if you can just make sure to silence your cell phones and computers throughout the day to minimize interruptions. So with that, I'm going to hand it over to Collin Hovinga, Vice President of the Rare and Orphan Disease Programs here at CPATH.
Thank you. Thank you again. Sorry, my voice from yesterday, I think I talked quite a bit, and so my voice is about gone. Just to orient everyone today, and we've got a lot of sessions. Yesterday we focused a lot about CP-RND and some of the initiatives around rare neurodegenerative diseases. Some focus on endpoints and whatnot, and then we had numerous breakout sessions. Today we're going to switch up the flavor a bit to focus a lot more on our real-world data platform and focusing on RDCA-DAP, which is under the direction of Alex there, who's sitting and looking at his phone right now. And then we have we're going to highlight some of our initiatives in patient-focused drug development with Tom Hart, and then we're going to talk about things that sort of lessons learned or de-risking things. And Andrew Lowe is going to give a talk about de-risking, but we're also complementing it with some a session that focuses on recent approvals, and then talking about the totality of evidence and package that was submitted, so that we can talk about the different strategies that were done for some of these recent approvals. So demystify some of the comments that we've heard, and as we've attended meetings, so people understand in rare disease really what it takes to continue to evolve a therapeutic development. And then the closing out of the session today, we'll be focusing on a great offering of hot topics, and we're excited to have people attend similar thing where there'll be breakouts. So just to start, I think our panelists are on now from Health Canada, and then EMA. We have from EMA, we have Christina Larson, and from Health Canada, we have Fiona Frappier and Andrew Taylor, and I'm going to allow the team to switch over to video for their presentations. This will be a live stream, so thank you all for attending.
Thank you, Collin. Should I start?
Yes, please. Thank you, Christina. Good to see you.
Yes, good to see you too. Well, I would have loved to be there in person, but at least, this is a possibility for me to share some views and experience from the European perspective. And I will try and share my screen here. And the presentation. I'm just trying to ensure that it is in presentation mode. I hope you can now see it all. Right, so maybe first of all, my name is Christina Larsson. I'm in charge of the Orphan Office at European Medicines Agency. And please let me know if you can't hear or anything happens, then I will stop and we can address it. But if you don't say anything, I'll just plow ahead. And I have a very small agenda because I'm talking about initiatives from Europe, mainly from the European Medicines Agency, but also some other initiatives at the agency or in Europe, across Europe. So this, and I hope that you can get the presentation upwards because it has a lot of links in it. So all these are sort of links that you can click on and get to that particular area. And I will address most of these, not in the exact order that you see it on the screen, but the European Medicines Agency has quite a lot of various tools for not only rare diseases, but many of them are used mainly for rare diseases or orphan products. So it's a little bit like a buffet of various things to choose from. And I will start out with just explaining that certain of these initiatives and tools can be used at a very, very early stage of development, while others come in a little bit later. But in the majority of the cases, it is quite early and can reach all the way through to marketing authorization and even post-approval help for developers of rare diseases. And these, again, are sort of the various aspects that I will go into a little bit more in detail. I wanted to start out with our engagement with academia. And it's because it's not necessarily super new, because we have been engaging specifically with academia for many years. Even when I worked in scientific advice quite a few years ago, we had special advice for academia and special fee incentives for them. But that has now been more formalized, and since 2017 we have this actual framework to promote academia and to try and translate the academic research into actual medicines for patients. And this is, of course, also not only for rare diseases, but what we've seen is that the majority of the cases that academic research groups are working towards, and when they come to us, it's generally for very rare diseases. And what we have launched rather recently is actually a special pilot for advanced therapies with really a lot of sort of extra love. And what does this mean in practice? Well, we have selected, the idea was to select up to 5 ATMPs, and they have all been selected now. And they will then receive, the applicants for these procedures will receive regulatory support. They have a dedicated EMA contact, and sort of on an iterative basis, we can help the company and engage with the company developer, the academic group developer, to try and ensure that they understand from the quality development, from the clinical development, and to ensure that at the end, the data that is delivered to the agency for approval is actually something that we can use as well. And I think because there's a lot of steps that can be difficult for academic researchers to know exactly what regulatory standards we have and comply with everything. And it's also financially good because it has sort of similar fee incentives as if you are a small and medium-sized enterprise, in sort of 90% fee reductions or even no fees. So I'm really intrigued, and as we have chosen these 5, it will be very interesting to see how this goes forward. Apart from that, we have something that is maybe not focused on products per se. Yes, you can come with a product, but in general, this innovation task force and the discussions with the innovation task force are more related maybe to sort of diagnostic tools, methodologies, technologies, maybe types of clinical trial, databases. It can be some preclinical evaluation tool, etc. So the idea with this support is really that we have something innovative, maybe something that no regulatory, well, at least we haven't seen before, and you're interesting to get feedback. So this is very much sort of a brainstorming discussion with the experts from the network of the European Medicines Agency, from all the member states. So we try and get the ones who are most into this particular field, the one you're interested in, into one room, one meeting, and trying to give advice and discuss how this can be developed in the future, and guide you also sort of further, or that the applicant further, what they can do and which steps they can take after this. This is really rather early on in the development, and very interesting, I have to say, to attend these meetings. We also have something which is very much product related, and it's called the PRIME scheme, or Priority Medicines. And the idea here, just like the FDA and Japan has as well, for certain products that you think are a little bit more promising, there is a big unmet medical need, and these products can hopefully address them, that they can be designated as prime products. And the benefits are that very early on, the assessment team, so the rapporteurs from those countries assigned, will be appointed very early on. There's a kickoff meeting with those rapporteurs as well, so they will know the product throughout the development, as opposed to normally when the rapporteurs for a procedure for marketing authorization sees the product the first time when it is applied for marketing authorization. So usually this is a meeting with the company and discuss sort of what, does the product have an orphan designation? Is protocol assistance scientific advice needed? Is there a potential? And they are all eligible for an accelerated assessment as well, which then may or may not happen. And why am I talking so much about prime? It is, again, not specifically for rare diseases, but if you look at the right-hand side of the screen, you see that almost all the products that have been approved under the prime scheme are orphans, so rare diseases. So this is really where companies usually can claim that there is a big unmet medical need. And therefore, I think it is a really good tool specifically for development of rare diseases. Now, something that is very close to my heart, because I used to work with this for many years, is obviously the scientific advice, or it's called protocol assistance for orphan designated products. So the protocol assistance is unique to rare diseases, while scientific advice is, but it's virtually the same, but it's just called differently. And the idea is that we give advice, the agency gives advice the CHMP, Scientific Advice Working Party, gives advice on any stage of development. This can really be from nitty-gritty quality aspects before you start non-clinical studies and throughout the whole development, even after a marketing authorization again. Scientific advice can be given. It is mainly given by the agency, but we also have the possibility to have parallel consultations with health technology assessment bodies. And we also have occasionally parallel advice with the FDA. At those cases, it is not the letter from the agencies is not one, but I have to, because I have been involved in quite a few, having this discussion at the same time is always really, really useful for sort of getting one conclusion from both agencies in the end, and one way to work forward. Because of course, the majority of rare disease developments cannot take place just in one country or in one region. You have to find patients across. And of course, specifically for rare diseases, for orphans, there's quite a lot of fee incentives as well. So if you are an orphan designated NSME, you don't actually pay anything for the protocol assistance. You may see that on the screen, it also says qualification of novel methodologies. And I have a separate slide on this because a little bit like the Innovation Task Force, it's not also possible to get a qualification for what in the beginning we thought were going to be sort of biomarkers. So if you are developing a targeted product, for example, and you want to have a diagnostic tool for that, then you can come for qualification of that particular novel methodology. But we've seen that this has now been much, much broader, and various methodologies can be qualified. You can, for example, qualify a patient-reported outcome tool. And usually this is quite early. We publish all the qualifications on the website, but it's not always that it actually results in a qualification, because maybe something needs to be done, and therefore we also have the possibility of having a qualification opinion and sort of a letter of support as well. So the agency will say, yes, this looks very promising. However, a few more things might be needed for the actual development. So if you're interested, all the qualifications are found on the website. And again, I guess those are also not necessarily only rare, but you will see if you look into them that the majority of cases actually relate to rare diseases. So I also wanted to, so that's majority what at the agency takes place. There are other things, of course, as well, but those are the ones I really thought were most important. And across Europe, there is a lot taking place. And only some of it is sort of course, centralized, but I've had many meetings and heard a lot of things going on locally, nationally, but also some across Europe. So I wanted to mention a few, which the agency per se is not necessarily part of. Sometimes we contribute, but it is not organized by us. And one of the really big things right now is what's called the European Partnership on Rare Diseases. And you can see that it's really prospective. So this is not something that is in one way specific for rare diseases, because these European partnership exists for many different type of areas. So it could be, I don't know, shipping or agriculture, for example. But in this particular case, it's focused specifically, this one's focused specifically on rare diseases. And this is really a partnership. So it's not the European Commission deciding exactly what is going to happen, but the various member states of the European Union have to come together. And the idea is sort of to pool resources and ideas in a European research program, sort of. And of course, the goal is then to improve patients' life, patients with rare diseases, mainly, yes, to be able to diagnose them for treatments, and also very much multidisciplinary research. This is a call, and it actually ends on the 19th of September. So the submission deadline for all the ones who want to participate is until the 19th of September, so very, very close. And right now, a strategic and innovation research agenda is being developed by a lot of experts from across Europe, contributing to objectives and ideas, aims, goals for what this agenda should look like and what are we going to put our resources into, efforts into. So this is not only pharmaceutical development, for example, but one thing that I think personally is important is also the education of developers, so ensuring that already at the research stage, everybody knows that if I want a product to come to a patient, there are certain steps I have to go through. So it's very interesting. We don't have it yet. As you see, the deadline is next week. And the research agenda is also currently being developed, but I'm sure that by the end of the year, I guess it should be published. So keep an eye out for this if you're interested. I also wanted to mention the European reference networks on rare diseases. And these are 24 networks, various type of rare diseases grouped, in order to try and get researchers, physicians treating, nurses as well, but everybody treating patients together and in a network in Europe. For example, from the north of Sweden, where I come from, liaising with someone from the south of Spain. And this is possible in this network. And together in this network, there is also this rare disease coordination and support action to try and create a platform for all of these groups to have research and innovation together. And there is work packages, seven work packages. I only listed 6, as you can see in the screen, because one is sort of more the collaboration part. But it is really interesting trying to get people working together, trying to harmonize data collection, for example. How do you collect the data, ensuring that patients in Sweden and in the south of Spain can be maybe compared in clinical trials, maybe they can be put in clinical trials together. So it's very encouraging, really interesting. It's a very vibrant group of people as well, the ones I've met so far, and very engaged. So there's definitely huge hopes, and I hope that patients will really benefit from this as well. Last but not least, I just wanted to mention a few of the innovative medicines initiatives that are specifically relating to rare diseases. And the first one on the screen here, the Screen for Care, is specifically to shorten the time for diagnosis. And actually, it was quite high tech, because it's not only sort of newborn screening, but they're also looking into various type of artificial intelligence to screen electronic health records in order to try and, but with the information from the health records, try and identify patients with a specific rare disease. So very interesting. We also have the RDAP. It's also trying to speed up development for advanced treatments. I find the EU Pearl quite interesting, because it tries to get patients much more into clinical trials, and making the trials more efficient and patient friendly. And they try and set up this clinical trial platform, so you can have multiple companies and multiple candidate drugs simultaneously, which turns out maybe not always so easy for a single company to do, but with this consortia, maybe it will be possible. It would definitely, I think, help patients, and they're very much part of it as well. And the last one I just wanted to mention for the IMI projects is this Connect for Children, which is a very large collaborative European network aiming to do clinical trials for pediatric populations. And there, of course, the majority are also rare. And there's both public and private partners in this, and they have more than 400 experts across Europe trying to identify and decide how to best do a clinical trial. It's a little bit like a one-stop shop. You can come to this group, that's the idea in the future at least, to come to the group, and they're piloting this and asking for help, asking for people to collaborate with, trying to define endpoints, trying to ensure that the best clinical trials are done, and ensuring that the patients get the most out of the clinical trials that are being done. So yeah, there's really a lot going on, and I only scratched the surface. There is loads more, but maybe for this morning, this will suffice. And there is a lot more interesting links if you wanted to have a look afterwards. And with that, I thank you very much, and will hand over to Andrew, I think. Thank you.
Thank you. Fiona, you go next. We'll take questions at the end.
Okay, I'll go first, and then Andrew's going to follow. Thank you so much. It was really interesting for me to hear everything going on at the EMA as well. Now, Tanya, are you going to share the slides for us this morning? Or should I do that?
I'll bring them up, Fiona. I've got them right here.
Oh, thank you so much. So while we're getting set up here, I'll introduce myself. Good morning, everyone. Thank you for inviting us. I know it's hard virtually, but we're happy to participate. So thank you even for a virtual invitation. My name is Fiona Frappier. I am a manager in the Biologics and Radiopharmaceuticals Drugs Directorate. This is part of Health Canada's regulatory branch, and I focus on rare diseases. And so that is my introduction this morning. Andrew, who I'll let present about halfway through, is a manager in the strategic policy branch and actually has a whole new national strategy on rare diseases. And so you'll hear both from the regulator today as well as from our strategic policy branch. All right, next slide. I think to begin is really to look at, a lot of the trends we're seeing, they're not national in nature, they're actually international. And so we're seeing every year more and more new active substances for rare diseases. It's basically half of new active substances every single year in Canada and elsewhere. We expect this to be a growing trend, partly because of how many new technology platforms are coming out, which are helpful to address rare diseases, but also our advancement in pathophysiology knowledge of existing therapies and diseases available. I think we've heard many times pediatric drugs continues to be an issue, in Canada as well as internationally. In Canada, most children do receive off-label prescriptions. We would like to address this, and that's going to require some continued clinical trials and innovation. We've also seen some great advancements in gene therapies. This is something that's been ongoing, of course, for 30 years, but as the learning increases, we're actually advancing some of the complexity of the questions that we're asking, and a lot of new data needed to address them. All right, next slide, please. So how do we tackle this in Canada? I think is pretty direct. We have, first of all, of course, clinical trials. In Canada, it's very simple to start launching a clinical trial. It's a 30-day no objection letter application. And so I would like to encourage everyone in the audience who's running a trial, in the United States or in another country to open a second site or an additional site in Canada. About 30% of trials in Canada are global in nature. And we welcome that. You can come in for protocol assistance. This is in the form of what we call a pre-CTA meeting, so a pre-clinical trial application meeting, and we can ask quality questions, clinical questions, and it can happen at multiple junctures as you come in for each additional phase of trial development. Once those trials are ongoing, we do like to see them registered. And so most Canadian trials are actually registered on the NIH site as well for clinicaltrials.gov, as well as the WHO site. We have a clinical trial database. I think this is very important to make sure we're leveraging the studies that are ongoing already and that we're making best use of those patient populations, and maybe where possible, even leveraging control groups. And so this becomes strategic in nature for us. Once we have data packages ready and we want to come for new drug submissions, it's a 300-day review pathway. Most rare diseases actually apply for 2 of our accelerated review pathways, though, and so I've highlighted those here for us today. The first is the priority review, and this is where we're really seeing substantive data, looking at an advancement over what is already on the market, or where there is no drug available for a severe condition. The other one I would draw our attention to for rare diseases, and we don't do a lot of these, we do about 3 or 4 a year. They're called Notice of Compliance with Conditions. They're almost always for rare diseases. And this is where we're seeing promising evidence. And so it is often coming in with one strong phase 3 trial where we agree that the data is strong enough to authorize the product, and then we offer a condition for a follow-up confirmatory study after the product is actually marketed. And so this can be submitted 2 or 3 years later to confirm the original promising results and further characterize the product. The package does need to be strong, of course, but for rare diseases, I think it is acknowledging that the evidence will be different than in some of the larger disease structures. And then the other pathway is called Special Access Program, and this is essentially a release program. It is very specific, and so a physician would apply on behalf of one patient for a release of a product that's not already on the market in Canada. They would need to submit evidence. It could be journal publications, it could be early in its nature, but this would be again for someone who's really facing some serious, life-threatening, no alternative situation where we think there might be something promising in development. And normally the special access program would only be a release onto the market, and so it would be a right to import. We've also developed within this something we call the OLIP program, and so this is an open label individual patient study, and it's intended to bridge the gap on some of the evidence needs for this program. And so this would be not only the release, but actually a small clinical trial, N of 1, maybe up to N of 10, to look at further characterizing why this would be appropriate in this disease condition. This can happen for quite a number of very rare diseases where they're looking for an advancement on a small level. All right, next slide. This leads us to our international collaborations. Canada is a small market. It's about 4% of global pharmaceutical sales. It's also about 4% of clinical trials, so that lines up. We work very hard with our international partners to make sure that we're coming up with regulatory strategies that are, enabling and aligned and offer, supportive metrics. And so the main way we do that is with the ICMRA. This is the International Coalition of Medicines and Regulatory Authorities. This is representing, I think, 60 plus countries. I've lost track of the exact number, and the regulatory agencies. We also work with, of course, PAHO and the WHO and their biologic standards groups, as well as the regulatory groups. But we also look at ways to align some of our regulatory work, and this can be internationally through parallel reviews or information sharing with the EMA or the FDA, also with our Access Consortium, which is with Singapore, the UK, Switzerland, Canada, and Australia. These are all kind of small, medium-sized markets where you can, as a sponsor, come in with one application and go through a parallel review. We would all have independent reviews within our own structures, but we would try and mitigate some of the regulatory questions and advance in a coordinated way, so it would reduce the burden on the sponsor. There's also some collaborative review programs through EMA Open, which is focused primarily on COVID, but is now expanding, as well as the FDA Project Orbis, which is focused on oncology, which is about 50% of rare disease new active substances in Canada every year. Also within this, there's a focus on post-market monitoring. And so a lot of rare disease products do come in with packages that require ongoing data support once they are on the market. And so it's important for us to think about strategic ways to leverage that data nationally and internationally to make sure that we're monitoring all of the best data available. All right. Which leads us to real-world evidence. We do real-world evidence primarily on the post-market, actually, but we're starting to explore how we can best advance it on pre-market strategies. And I think this, for you, will be very familiar. It's really ensuring that it's grounded in data integrity principles, which would apply to any data package that's being developed. But we've looked at ways to create position papers, guidance documents, and really some key instructions on how to optimize that in a way that will lead to stronger, more reliable data sets as they come in to support endpoints or supplementary endpoints for new packages. Next slide, please. And then within this, we're actually in an innovation cycle. In 2019, the government came out with a large budget announcement on health and biosciences and looking at novel ways that we can increase biomanufacturing in Canada, but also ways in which the regulator can improve its approaches. And so there's been 4 key pillars for this, all of which would apply to rare diseases actually. So modernizing clinical trials, this is looking at how do we better enable decentralized trials, how do we make sure that we're improving monitoring and data collection. We are also looking at enabling advanced therapeutic products. And this is a bit of a sandbox approach for the regulator. And so when we're really looking at exceptionally innovative approaches, maybe cell-embedded medical devices that would then be implanted in organs, that we're creating regulatory clarity on what we would need to see to advance these products through to the market. And so these products can come in and receive kind of a white glove concierge service that will help enable them through those more complex regulatory packages. We're also looking at agile licensing for drugs. And so this is looking at enabling life cycle management through rolling submissions, as well as terms and conditions, and ensuring that we're modernizing our concepts on risk management plans as well for marketed products. And then lastly, but importantly for rare diseases, when we think of diagnostics, we also want to make sure that we're using agile licensing for medical devices and advancing our regulatory pathways. And so this is a 5-year program to review and update all of these regulatory mechanisms. The other very important piece I would like to bring up, and I think many of you already know this, but it's looking at transparency in communicating with all of our health system stakeholders. And so we do this in many different ways online. The first is our submissions under review. We often get asked, we hear the FDA is reviewing this new product, are you reviewing it as well? So all of this is published online. It's on our database called Submissions Under Review, and people can find the date it was submitted to us, what the actual indication was that's being asked for, and track it, in real time. We also publish the regulatory decision summaries, and this is where we can look at the technical advice once that's come in. So what is the clinical basis of decision, as well as the quality review for that product. We also offer a higher level summary called a summary basis of decision. This is meant for a broader audience where we can look at what was, the inclusion criteria, the exclusion criteria. Why does this indication come through the way it did? And then within this, you can always look up through our drug product database, what are all of the marketed products in Canada? What is the indication that it's authorized for? We do see quite a bit of off-label prescribing in the rare disease space. This is normal, but we would like to see some advancement on improving labels where possible. And then lastly, we also do a clinical release of the information. And so this is the clinical data that was reviewed to support the package gets released as part of our public release of clinical information. And it's an online portal. This is done in collaboration with the companies themselves. And actually, it should support further advancement of our understanding of clinical packages and what can be done for next drugs as well. And I think with this, I hand it over to Andrew to talk about our national strategy on drugs, and we'll take questions afterwards. Over to you, Andrew.
Yeah, thank you, Fiona. So my name is Andrew Taylor, and Fiona works in our regulatory side, looking at the safety and effectiveness of drugs, or efficacy of drugs. And then I'm over in the more strategic policy branch side. So I'm looking at pharmaceutical policy writ large. And so I'll talk a little bit about a recent strategy that we've released related to drugs for rare diseases and trying to support various aspects of their development and getting access to patients. So you probably know this already, but Canada has a complex pharmaceutical management system. Fiona's talked quite eloquently about what Health Canada does for authorizing drugs for sale, looking at whether a drug is safe and of suitable quality, and making sure that it's not of excessive price through our patented medicine prices review board as well. But there are other aspects within the system. There's other decision making. There's other data needs. So we also have a health technology assessment body working at the national level and in the province of Quebec, looking at evaluating the clinical benefit and the cost of drugs, trying to get a sense of cost effectiveness and value for money. We also have a Pan-Canadian Pharmaceutical Alliance. That is a collaboration of provinces and territories working with manufacturers to negotiate prices that are more accessible for the system and for sustainability of providing care to patients. And then following this HTA and pricing negotiation work, drug plans typically then make the decision about whether to list a drug on their formulary. And in Canada, we have both public programs through our provinces and territories, as well as private plans, and each is making decisions a little bit differently. So this can create a postal code lottery, if you will, in Canada, where based on which jurisdiction you live, on which kind of access to private or public plans you have, patients have varying access to drugs for rare diseases. And we heard that that's a major issue that needs to be addressed. So the culmination of this was really a broader advisory council on the implementation of pharmacare generally in Canada. And they noted the importance and the need to develop a strategy specifically for drugs for rare diseases, not just because of the cost associated with it, but because of that postal code lottery and varying decision making across the country. So in budget 2019, the government allocated what is essentially $500 million a year to support a strategy to try to improve access for patients. So starting in December 2020, the federal government came together with our provincial and territorial colleagues, established a working group, and tried to look towards how we could develop a national strategy for Canada and how we could build more consistency within decision making. So we held broad stakeholder engagement between 2021 and 2022, supplementing the work that we were doing in provinces and territories, reaching out to manufacturers, hopefully many of you here today, researchers, patients, of course, clinicians, and other types of people with a stake in trying to improve the situation. And we have a What We Heard report and various discussion pieces that sort of support our thinking on that strategy. But ultimately, the culmination of that work and that consultation, there was a vision and a series of pillars that really emerged from that work. And really, it's focused and the linchpin of it is providing better access to effective drugs and thereby supporting better health outcomes for patients in Canada. So we're guided by 4 pillars that really focus on patient outcomes and sustainability for the system writ large, working towards national consistency and trying to make sure that people have the same access no matter where they live across the country. Investing in innovation and research, because we know that that is super important for the rare diseases where we still don't know a lot about natural history of the disease or even have health products available to treat those that are living with those conditions. And also understanding the importance of data infrastructure and supporting the collection and use of evidence, which my colleague at the EMA spoke eloquently to as well earlier. So ultimately, this framework really led to the announcement of our strategy back in March 2022. And we've tried to have a series of activities associated with these various pillars. So to support patient outcomes and sustainability, we're working with our provinces and territories as well as Indigenous Services Canada at the federal level to make sure that we're improving access to new and emerging drugs, those drugs that are potentially challenging the system, either because of cost pressures or because of a new way of doing things and a need for better harmonization and standards and access across the country. But we also know that we need to enhance the existing drugs that are already on public formularies to try to make sure that each formulary is brought up to a minimum standard and that there's better access across the country. But we also know beyond drugs, enhancing screening and diagnosis was mentioned as a major issue during our consultation work, making sure that we're applying novel approaches to screening and diagnostics and helping to get patients earlier access when needed. So we're currently working with our provinces and territories to create agreements to support these 3 areas. in the effort to support better patient outcomes. And then to seek national consistency, we're establishing a few governance structures at the national level. So we're continuing the working group in a more formalized way with our provinces and territories, but also implementing or establishing an implementation advisory group. And so this is a way for us to continue to work with and consult with patients, manufacturers, private insurers, and other types of stakeholders that worked with us during the consultation work to develop the initial framework for the strategy. But these last two areas are what I'd like to talk a little bit more about. I think what you'll be most interested in, which is what we're doing to improve the collection and use of evidence. So we're working with our partner agencies, our health technology assessment body in Canadian Agency for Drugs and Technology and Health, and one of the major data institutions that holds a range of administrative databases in Canada, the Canadian Institute for Health Information. But then we're also working with our national research body, the Canadian Institute for Health Research, to advance a rare disease research agenda. And so on this slide here, you'll see the range of activities that we're working with. With CADF and CIHI, we're releasing a range of activities along the product development lifecycle to support various stages of decision making and the varying data needs that come not just from the regulator, but from the HTA bodies and the public drug plans and other people that are making decisions within the system. And then with CIHR, we're launching 4 funding opportunities to support rare disease research. So in the early research and development stage, we're developing a pediatric clinical trial network to better support trials across the country and enable those trials in children, with children. We're also supporting a funding opportunity related to clinical trial readiness, bringing that product to a stage related to drug development. And we're working with CADF to better understand what's coming down the pipeline through a horizon scanning type initiative to better understand how the system might need to adapt, either through changes in pediatric screening or newborn screening or other types of activities to support the eventual introduction of products within the system. We're also working with our regulatory and HTA bodies to improve the business intelligence around the decision-making process. So we're working with CADF to better understand disease-based registries in the rare disease space as part of the real-world evidence work that Fiona briefly mentioned. CADF is continuing that work of assessing what registries are out there, the quality associated with them, in an effort to build more standards and essentially a harmonization process of the data that is available within those registries to eventually get to a point where we can build the capacity to reach those standards in Canada. CIHI is also working to develop a drug plan and a formulary tool to understand better coverage decisions across the country. And we're working with CADF to develop in their review capacity to be able to look at novel drugs, potentially in a more innovative or novel way beyond the traditional kind of HTA approaches. And then in the screening and diagnostic space, we're working with CADF to better understand the newborn screening programming across the country in an effort to build best practices and consistency in that space, and also supporting CIHR to support sort of novel developments related to genomics and its use in the diagnostic process, potentially earlier in the process, to reduce that diagnostic odyssey for patients. And this really builds from other initiatives supported by Genome Canada in that space. And then in the final, once the drugs are to people, we want to be able to continue to monitor that ongoing use. So CADF and CIHI are working together to look at linking the administrative databases that CIHI has and the information and the wealth of information that's there with the registry information that we're looking at in the real world evidence space. And we're also working with CIHR to better understand how we can look at the administrative databases that we have to answer questions related to optimizing the long-term health of patients. So the next stages for the strategy are really to work with our provinces and territories to advance a small set of new and emerging drugs that could be cost shared and covered in a consistent way across the country. And that's one small element of the agreements that we're coming up with PTs. We're also working to develop consistency in screening and diagnosis, as well as supporting coverage for existing drugs for rare diseases. And then we're also continuing to work with patients, clinicians, private and public drug plans, the pharmaceutical industry in an effort to implement the strategy. And so in the coming weeks, we're proud to be announcing the establishment of our implementation advisory group and what they'll be doing to better support the next stages of the strategy. But of course, this targeted investment in rare diseases really just supplements and what is a broader wealth of work going on at the national level to be able to support drugs for rare diseases, as well as drugs more broadly that are innovative and useful and needed and meeting unmet patient needs for, across the country. So Health Canada does have a pediatric drug action plan in place with a series of activities to try to improve and encourage the number of drugs coming to Canada with a pediatric indication and trying to incentivize that kind of research. We also have the biomanufacturing and life sciences strategy, which came out of COVID, but is now reaching a much broader range to try to support cell and gene therapy more broadly, trying to develop the capacity within Canada for these types of therapies, as well as the research and development associated with them. My division is working to stand up a Canadian drug agency and trying to support national pharmacare. So we're looking to create legislation specific to pharmacare in the very near future, as well as what the functions of a Canadian drug agency could be to better support appropriate use, data and analytics, and pricing negotiation within the country. And then we have a pan-Canadian genomics strategy, which again is looking at the use of genomics within various aspects of various industries as well, health being one of them, and how we can better support that. And health data is currently being worked on as well at a national level, in addition to looking at pharmaceutical data specifically. And then through the National Research Council of Canada, we also have some very interesting challenges programming related to cell and gene therapy, and trying to better design and develop those technologies within Canada. So there's some links to all of those things. Can't obviously speak to them, but really trying to support rare disease and research generally to build access to these products for patients and to create Canada as a place where manufacturers would like to bring those products. So that's it for my slides, and Fiona's happy to answer any questions you might have.
Do you have any questions from the audience? I have one. I know that you all have touched on this a little bit in your slides, but I want to, realizing, that rare diseases are a global, issue. How do you all work well together, and how do you connect on strategies to advance drug development in various peds rare diseases? Because you mentioned peds quite a bit. I can inject that, but rare diseases in general, but peds as well.
Who goes first?
Christina, you spoke up. You're first.
They
said not it, so it was.
Okay. I was just now thinking, because actually we work quite closely together, and one of the things that we have is that we have what's called clusters between the agencies, and we do have one specific on rare diseases, and this cluster is between the EMA, FDA, and Health Canada. So once a month, we have a call, discuss various topics. It can be a product under marketing authorization. It can be discussions on some advice given, or indeed methodology to be used. So I personally find those very interesting, and it's also really, really beneficial to hear colleagues' views on how to best do the development and what is also acceptable in the various jurisdictions. So that's one possibility that we have, for example, to work together.
Yeah, the clusters is a great one because it's a very deep dive into the data. I think the other one I would mention is we work together on ICH, and so it's the opposite approach where we go to the principles that we can all agree to, and we write international guidance documents. And so ICH is the International Coalition for Harmonization and Regulatory, never going to get that acronym right, but essentially it does 3 levels of guidance documents, one focusing on quality, one focusing on safety, and then one focusing on efficacy. And so each time there's a new leap forward, say if it's statistical analysis and how do we want to handle Bayesian reviews of data, they'll write an ICH statistical approaches document, and then this can become principles that all of the regulators, EMA, FDA, Health Canada can agree to, and so it helps ground a lot of our work moving forward into those deeper dives as well.
Thank you. For sake of time, we'll, if there's any questions, we'll go to the next session. But I really wanted to thank each of you for participating in the session. I enjoyed our conversations recently, and I myself will be following up with you to further conversations about collaborations. Thank you.
Thank you.
Yeah. Tom?
Have a good meeting.
Hey everybody, we'll get started with the panel. We have Soren's coming up now, so we're all set. My name is Tom Hart. I'm the Director of Outreach for CP-RND at Sept, and we have a august panel, so I'm not going to take long to introduce myself. Nice to see you all. Thanks for coming. Before I begin, though, I do want to take a second and recognize, I think this is really important, that we're going to have this discussion about patient-focused drug development. There aren't a ton of people with lived experience here with us today, and I'm glad that there's a number of people who are able to join virtually through the live stream. They'll also be participating and asking some questions. So I'm glad that you're here. If you're watching virtually, thank you for joining us, and just know that we're committed to continuing that engagement and access in the future. So to begin, I'm going to ask everybody on the panel to introduce themselves and provide some opening remarks.
Good morning. My name is Shana Dodge. I'm Director of Research Engagement at the Association for Frontotemporal Degeneration, or AFTD. And for those of you who may not be familiar with FTD, it's an umbrella term for a group of closely related adult-onset neurodegenerative diseases. We don't right now have any approved symptomatic or disease-modifying therapies. But in the last few years, we've seen huge growth in the clinical research space. We now have a number of trials in phases 1 through 3. So we're feeling really, really optimistic, and we're looking for ways to convey that optimism to our community, many of whom were told years ago, you have this diagnosis, there's nothing that can be done. So we're working really hard to communicate that the narrative has changed a bit, and that in order for these trials to succeed, we need people to volunteer. We know that one of the rate-limiting factors in these rare diseases is people who are able and willing to participate in research. So we're always looking for mechanisms to understand our community so we can understand those barriers, we can understand the lived experience. And we were really lucky in 2021 to host an externally led patient-focused drug development meeting. And in conjunction with that, we deployed a large community-based survey that touched on many of the topics that PFDD meetings touch on, like the diagnostic journey, what symptoms are most distressing. And between those two initiatives, we now have a beautiful portfolio of qualitative and quantitative data that we can now share with those that are interested in understanding how to connect with our community. And one thing that we are always looking to do is really create a sense of community, because FTD is clinically very heterogeneous, and many of the current trials are targeting a very small subset of our community, those that are impacted by a specific gene. So we're always looking for ways to ensure that everyone understands that progress in one domain is really progress for everyone. And we also know that there's a lot of work still ahead of us. We just heard all about how things are looking on the international space, and we know that for clinical trials to be successful in FTD, they need to work globally. So we're trying to better understand how we can support international clinical trials. And we also know that we're only hearing from a small sliver of the population when we're doing things like PFDD meetings because of inequities in access to diagnosis. In FTD, there's a lack of biomarkers. So we're constantly looking for additional mechanisms to hear from our community and hear from those who have been historically excluded from the narrative. So I'm really excited to be on this panel and learn how others have been successful and how they've overcome some of the frustrations in this space as well. Thank you.
Hi everyone, I'm Ed Nealon. I'm a pediatrician and medical geneticist. I'm also the Chief Medical and Scientific Officer at NORD, the National Organization for Rare Disorders. I've been a physician at Boston Children's Hospital for 25 years, and I still see patients there part-time. I also served in the past as the head of global medical affairs for rare neurological disorders at Sanofi Genzyme and worked on 4 clinical development projects there. I counted it up the other day. I guess I worked on 16 different clinical trials and 7 different rare disease drugs that have been approved by the FDA. Obviously now, for the past 2 years, I've been focused on activities at NORD, but I like to say that really my credibility as a patient advocate goes back to work with smaller groups for a longer period of time. Since 2006, I've been the principal medical advisor for a very small patient advocacy organization for a disease that has an estimated incidence of about 1 in a million and can never quite get to the point of having clinical trials or anything done for them. I'd like to touch on a few things in opening comments. I'm looking forward to hearing all the perspectives on airing the voice of patients. NORD has historically, through a memorandum of understanding with the FDA, played a role with the development of patient listening sessions at the FDA. And we also help organizations, usually NORD member organizations for representing single diseases, to conduct PFDD meetings, most recently one for galactosemia. But there are a couple of issues beyond just how do we air the opinions of patients and get those integrated into development plans earlier. With an estimated 10,000 rare diseases, many of them affecting very few patients, like the condition I discussed. It's a reality that 95% of them have no FDA treatment, and many have no real substantial effort underway to develop drugs. NORD has some programs to help new patient organizations form so they can begin to advocate for their diseases, as well as some educational materials and other programs to help patient advocacy groups try to make their disease research ready and attractive for pharmaceutical development. We have a new effort, which is really the reason I was hired by NORD, which is the designation over the past couple of years and now the coordination of a nationwide network of clinical centers of excellence, which we hope will work together and make it easier to study diseases, especially those with very few patients scattered across the country. And another sort of elephant in the room besides those diseases that are really getting too little attention and being left behind is that data sometimes gets siloed. And there I'd just like to mention a key collaboration between NORD and CPATH around RDCA-DAP. NORD is a believer that since patients and their advocates have the greatest incentive to share their data, that it is a good and useful thing for patients to own that data. We run a registry and natural history study platform that about 46 different studies are hosted on for about 40 different diseases. We're encouraging as much as possible that data to be shared with the world through CPASS, RDCA-DAP, and I'll stop there.
Thank you. My name's Kathy Collin, and I almost gave Ed a huge amen. So that was about getting us out of silos. I'm Kathy. I'm from Indianapolis, Indiana. We lost Mom to ALS in 1997, and I found it outrageous at the time that her prognosis was largely the same that Lou Gehrig had in 1939. And it's all the more outrageous for people dealing with ALS today. My career path has very much influenced my advocacy path. I worked for Eli Lilly and Company for 18 years in the late 20th century. And then I was one of the principals at a small consulting practice for 23 years. I retired a few years ago and have more recently been described as a retirement failure. So I'd like to talk to you just for a few minutes about what people with lived experience have for you. They have gifts for you. If you are a government organization or a drug company, they have huge, valuable gifts for you. And you get to pick the kind of gift you're going to get. Now, some of those gifts will be the gift of empathy. They'll tell you their stories, and you will become much more understanding and empathetic. Some of them will give you the gift of loyalty and gratitude, where you tell them what you're doing, and they will be just very, very supportive of what you're doing. But I maintain that sometimes the most valuable gifts are the hardest ones to open. And I say, look for the good troublemakers out there, because they are going to give you gifts that will make you better. And that's not always easy to do. One of the great good troublemakers that God put on this earth for a short period of time, because she had ALS, her name was Sandy Morris. She used to pass out nice key fobs that would say, what you allow is what will continue. If you Google Sandy Morris, the nation, you'll see a wonderful tribute to her. We lost her last year, but she was one of the great troublemakers and accomplished so much in making clinical research better. Now, once you identify your good troublemakers, how do you work with them? And I have two messages that I'd like to send to you today. One is, make them part of your project teams. Integrate them into the work. Don't go do your thing that feels all good and patient-centric and then bring it to them and say, how do you like it? Integrate them, give them work to do. Make them have deliverables too. And I guarantee it will be productive for everybody on the project team. The other thing is, one thing I've noticed over the years, you folks pick awesome people to be your patient liaisons. They are lovely people. They're usually kind, empathetic. HR does a great job at finding the people person who's just right to do that job. But I beg you, give them some juice. If they don't have the attention and interest of top executives at your organization, then what we're doing is just patient-centric theater. You really have to give them a way to help the people they work with make a difference. So those are a few thoughts for the day, and I'm going to pass it along to somebody who's kind of a legend in this area. So, Pat?
So amen, Kathy. First of all, I'm Pat Furlong. I'm founding president of PPMD, or Parent Project Muscular Dystrophy. We're going to be 30 years old in 24, so that means like when I was about 15, I decided I'd look at Duchenne. So the real story is that I come from a background of master's degree in nursing and a nurse practitioner, and I spent my time in whole organ transplantation and renal dialysis, and then ICU and CCU. And it was all adults because I thought children should not be sick. And I still have that firm belief. On a very cold day in June in Middletown, Ohio, my son was trying to ride a bike but couldn't. And we were worried about these two little boys that we had because they couldn't ride bikes, they didn't do stairs. But when Chris attempted to ride a bike, I think he tore his Achilles tendon or injured it. He cried out in pain. We took him in the house. I slept with him. And in the morning, my husband was in tears. He was in medicine until he retired, which is a different story. I asked him what he thought was wrong, keeping in mind I'm all adults, so I recognize these boys were delayed in terms of they didn't act like other little boys, and I was completely justified to say I was the best mother with the most well-behaved children, which turned out to be a lie. And when I met with a friend of mine, an orthopedic surgeon, he said, this is Duchenne muscular dystrophy. And he asked me what my younger son looked like, and I said the same body and the same delays, and he said, you have two. So I went down to Cincinnati Children's Hospital. I saw a person to confirm the diagnosis, and he said, there's nothing to do here. They're going to die. And I said, okay, so just fundamentally they have flat feet. So if I put an arch under their foot, would that improve their gait and maybe extend their gait? And he said, who cares? They're going to go off their feet, so why bother? So it took me some time. I went around the country and around the world to figure out what do we know, where's the money, who are the people, what are we looking at known medical tools, and I started PPMD. So Duchenne muscular dystrophy is an X-linked disease, primarily happens to males, although some females do have symptoms, and carriers can have some cardiac problems and also some weakness. I ran around the world to find out where we were and decided that patients and families are the best gifts to this community, and they're the ones, the troublemakers, that are going to make a change. We started PPMD 30 years ago. We started building our tools by asking what medical tools can we use to implement or change the trajectory of this disease. We started with cardiac medicines, physical therapy, steroids, which were tested and tried and thrown away, and we now have about 40 years of steroid data. And then we began to build other tools about if we invest money to fully characterize the disease, where will that take us? If we leverage NIH money to improve that characterization, where will that take us? We began lobbying the federal government for money, for interest, and that led to a tipping point of industry. So patient-focused drug development is where we have a collaboration with the patients, with federal agencies, with the NIH, with industry, and with payers. Because dragging parents through this process, they're going to be able to tell you what their preferences are. So we know what the priorities are. We know as these young men get older what their preferences are. We work with FDA. We've written draft guidance for FDA. We have another draft guidance that we just completed as we're in the area of gene therapies. So we now are happy to say we have 4 approvals in antisense oligonucleotides, and we are now in the area of gene therapy with an approval on June 22nd, and now 20 children infused with the gene therapy. It's a great deal of uncertainty, but our problem right now is those children that are now receiving a gene therapy, how are we going to do this combination therapies for those children? And I don't mean the cardiac meds and the steroids, I mean disease-specific medicines to protect whatever is delivered by gene therapy. So we're in this world of patient-focused drug development where we have to wrap our arms around this new era of gene therapy and figure out a way to diagnose early with newborn screening, to have a registry so that we are collecting not only the real-world evidence, but we're pulling in that electronic medical record so we can understand what gene therapy is doing for these children, protect them, and add on new medicines. So I agree with you. We need troublemakers, and patients and families are not just the checkbox for industry. They are the guiding light for what trials look like, what evidence is needed, what real-world data looks like, and what payers will need to be able to cover the cost of these drugs. Because it won't help if we develop them and we can't access them. So PPMD is still on the road of trying to really solve these problems for Duchenne. Next. You're next.
Well, good morning, everyone. My name is Michelle Campbell, and I am the Associate Director for Stakeholder Engagement and Clinical Outcomes in the Office of Neuroscience in CEDAR. I am about to celebrate my 9th anniversary at the agency, and I began my career at the agency. Actually, I'm a measurement scientist, so I was originally in our division of what is now our Division of Clinical Outcome Assessments. And so in that role, they are internal consultants to the review groups, and I worked across a lot of rare diseases, providing advice. And about 4 years ago, I transitioned specifically to neurology, which is where I had spent some time in my prior life, to the agency in a neurology and developmental medicine department at a hospital. And so, and Pat happens to be one of the last people I saw in person pre-pandemic. And I'm so happy that she's next to me because she has been a great champion and someone that we do collaborate with. And I think she highlights the great new unknown world of now having options, you know, in therapies. And to Kathy, I like to search for trouble personally myself before trouble finds me to find solutions. So what does patient-focused drug development mean to me, and how do we really work with it every day in our life and in the agency? So, you know, when I joined the agency 9 years ago, we were in the infancy of PFDD, right? So these patient-focused drug development meetings were just happening under PDUFA 5. And I kind of thought it was weird. Why weren't we already talking to patients and caregivers? Because I had come from a rare disease pediatric side, where every day in clinic, the clinicians, that's what they did. They talked to the caregivers about what's going on in their child's life to help make informed decisions on their care. So to me, I'm like, don't we always do this? Why do we have to make it special? And I always thought, you know, we need to learn from our rare diseases because they do this all the time. And so it has been it's been fabulous to be part of the PFDD effort at the agency through its growth over the last decade. And it has seen massive expansion and growth during this time. just starting to listen to patients in those meetings to now where we talk about how do we incorporate patients throughout the life cycle of a medical product development. It's not a one-time, what is that endpoint, or what are those things you want to measure, but where are those opportunities? How is the agency just one stakeholder that should be part of the table in the discussion of optimizing clinical trials? And we have launched successfully our externally led patient-focused drug development program meetings, which if to know how much we are dedicated to this, in 7 days, this is my 3rd rare disease meeting I've been at, and I end my week at another one. So we are very much committed. So when we can be able to participate, we oftentimes want to be the listener in the room. We want to learn from these experiences. We take that information back. We take that back, and when we have those INDs come in for review, those protocols to review, we think about those conversations we've heard. We go back to those voice of the patient reports and read them again. Patient-focused drug development and coming and talking to us is not just when there's a marketing application, okay? It is throughout that entire drug development process. And so sometimes it's really hard to be able to see a tangible thing from what we learn immediately from these meetings, because we have to wait for something to come in for us to review through that drug development process. But do know that we do take into account, and it's more than just us acknowledging a checkbox when we get a marketing application where we have to say, was patient experience data contributed? It is really our everyday life. Patients are in the forefront of our mind when we make our decisions. When we are providing that advice, we are always thinking about, is this too much burden? Am I getting the correct information to help inform? Am I measuring the right information that's going to help us really be able to see, well, can we support efficacy, and of course capturing safety, which is critically important. So it is, to us, our everyday. This is part of our job. This is what we do, and our patients are always in our mind. I think it's an exciting time in rare disease. We're continuing where over half of new molecular entities every year is for a rare disease. But we have many more to go, and we know that. I've been saying the last couple meetings, I wish I didn't have my job. Please kick me out of my job, right? Where we have treatments and cures for our diseases. Reality is, I'll probably still have my job, right? We still have to monitor and make sure we have good data. But it is our everyday life. So this is why we talk about engaging us early in those conversations in the drug development program, as well as finding the opportunities to engage with us through non-drug development programs to really learn about what is that lived experience. That's what I want to know. I want to know what your lived experience is. So I want to thank the patients and the caregivers and loved ones who are here in person today and that are online for taking time to participate. And hopefully, that we are, there's a whole team back working at White Oak right now, trying to do what I just described every day, because this is our everyday.
Thank you. Hello, everyone. First of all, grateful and humbled to be here, and want to thank everybody on the stage for their efforts and their careers to advance PFDD efforts. My name is Serin Fidelis. I'm the Executive Director of the Polycystic Kidney Disease Outcomes Consortium. And while you guys heard yesterday of our efforts to develop tools and disease progression models in the ADPKD space, we have in the last year focused on the recessive form of PKD as a case study for putting together an externally led patient-focused drug development meeting. And this was in talking with our stakeholders, in talking with our FDA liaisons, obviously patients. The need for this effort was apparent. ARPKD is an ultra-rare disease. There's 1,500 to 2,000 patients in the US affected by it. Again, it's very heterogeneous. It's got the early presentation with pulmonary hypoplasia, with high mortality, and then patients who make it past that perinatal stage have the kidney phenotype, so this rampant cystic kidney disease that leads to kidney failure in their teens, early 20s. And then there's the liver phenotype, the congenital hepatic fibrosis that is associated with portal hypertension, esophageal varices, splenomegaly, et cetera. So, complex disease, huge unmet need, no real pipeline for it. So we wanted to take a different approach and, gain a foothold with respect to the, patient perspective and patient journey in ARPKD. So we held our meeting a couple of weeks ago, really, it's the comment section is still open for patient to leave their comments about their lived experiences. So, if anybody knows any ARPKD patients, please spread the word. But again, the meeting was really powerful, moving, and really important because we collected a lot of information. We had surveys, qualitative outputs, and as Michelle said, this resource doesn't live in a void, right? It will turn into a voice of the patient report. It will be part of, future, essentially evaluations for drug products in the space. So, the importance of having this cannot be underestimated. And, it was really interesting to, for the clinicians in the audience, it was a virtual meeting, to hear about, symptoms and burden of disease that they weren't aware of, right? And that is why having the patients at the forefront of these types of efforts is just, critical. And, we hope that this is the first step with respect to, building future PRO tools for ARPKD, and, having those as part of, future, obviously, drug development efforts and composite endpoints for ARPKD. Again, very grateful to be part of this effort. This was done together with the PKD Foundation, and we work very closely with James Valentine, who used to be at FDA and now advises PFDD efforts. And again, it was a very collaborative effort, and we really hope that this is the stepping stone to continued efforts in this space with respect to the incorporation of patient voice in drug development efforts. So, thank you.
Excellent, thank you. In listening to these comments, there were so many good questions I want to ask. So let's start with Kathy. I know we talked about this a little bit. Bringing together stakeholders from academia, industry, and advocacy, people with lived experience in these different backgrounds, there can be some gaps. So how do you speak to unmet need and approach those gaps that you've identified? And everybody chime in, but we'll start with Kathy because we already talked.
Wow, okay. So there are gaps, and we find them all the time. ALS is a high turnover disease. You have to realize that. A lot of our best and brightest get ALS, but they're not necessarily familiar with regulatory affairs. They're not necessarily familiar with neuroscience. So it's like, but we find in ALS land that there are people who do a lot of studying. I mean, they'll really take the time, read papers, try to understand them. And I think that's one of the reasons why we need to pull people together and do more listening than we probably do. Yesterday, I know I learned a lot on the regulatory front. Just, and anybody who's been paying attention to ALS knows the term regulatory flexibility has been bandied about like a sponge. And I learned things yesterday about the nuances of regulatory flexibility and what FDA looks for. And we need to do more studying like that. But we do, and there are people in ALS I admire greatly who take the time to learn. This fall, or actually December, a group of us volunteers, were sending 8 people with ALS to the largest scientific symposium on ALS MND in Basel, Switzerland. And there was an application process. We couldn't accept everybody, but there are people who really want to learn the science, they want to learn the government perspectives on things, and they want to learn the practical clinical issues. And they're out there. We just need to look for them and help them get that information. Thank you.
Excellent. Anybody else want to chime in, or I can move on?
Sure. I think that when, in the rare disease community, when you're diagnosed or your child is diagnosed with a rare disease, you want to become a citizen scientist by default, right? Because you have to know, you have to find the physician who's willing to provide the kind of care, the physician that you're going to trust. You have to find others so you can learn from them in that, you know, what happens next? What did you do in these circumstances? So I think you become this citizen scientist, and then gathering you together, their willingness to share knowledge, to contribute to a registry, to participate in the patient-focused drug development meetings, they want their voice heard. They want to be seen so that together they can make progress. There's nothing like participating to deliver hope to your family. So these families are actively engaged. So we hold conferences every year. We have a registry that's now pulling in that electronic medical record. These are things they want because they want their data out there in the event that they could participate in a trial or they could access a therapy that's approved. And I think the other thing is when we first had met with the Office of CEDAR and Michelle and Dr. Dunn, there are sometimes patients feel like that is this black door that they can't go through, right? And walking through there and sitting down with human beings that are in the FDA that sometimes you don't think, well, they're not human, right? You can't cross that line. But to talk to them about, we want you to understand our disease. We want you to understand what's important to us. And for example, the 6-minute walk test was mentioned yesterday. We hated the 6-minute walk test because we don't care if our boys walk 6 minutes. Walking from a chair to the table, lifting their hand to their mouth, that's what matters, not 6 minutes. So then we had to work together to understand what are you getting out of this and can we modify it? We now have a North Star assessment, which is 17 measures, and Joanne mentioned that there's a monopoly and there's iterator reliability and all the good stuff. But the fact is, these are 5-year-old boys who don't care about 17 measures. So now we look at places like CTAP and say, could we look at those measures and modify them and get 4 or 5 that are predictive of that 17? So this is collaboration, I think, and then going to the FDA and say, hey, not the North Star, but could we do something different? And participating in trials, 4-year-olds in Duchenne and ALS, none of these patients are alike. So do you recruit by age or do you recruit by function? So these are where patients and families want to contribute, to hear their voice, to actively participate, and certainly to be the recipient of therapies and combination therapies like we all hope for.
Well, Tom, I'll add one thing. There are a lot of gaps, and so it's hard to make a general statement about all the gaps, but I think an important element to emphasize here is that if a gap is identified, be it by a patient or a parent or a physician or anyone, look for a way to turn it into data so that you can change people's opinions at FDA companies and so forth. And I mentioned before a very rare disease where I've been supporting a patient group for close to 20 years. It's a horrible disease where most of the children die in adolescence. And yet, one parent says, my kid's skin is itchy. It's not in any of the textbooks, right? But it ended up taking a sort of survey. It turns out, nearly everybody's kid's skin is itchy. And so is that a peripheral neuropathy? Would it be responsive to drugs that are used for peripheral neuropathies? It only starts to close the gap if you find a way to turn it into data.
Yeah, Tom, I hope people take into account the folks that are on this panel represent the diversity of rare disease, from pediatric to adult, slow progressing to fast progressors, right? And each individual has their own unmet needs. So there's 7,000 unmet needs that we have to face. And so I do think that one thing that I want to highlight was gathering that information to come have a conversation with the agency or informing a drug developer on here's what you need to know. Because we can only react to what's given to us, right? I can't react to something that I don't see, right? So how can you leverage that? But also, I think what Kathy highlighted, and I know that Pat has experienced, is not everyone may be able to participate in a trial, but there's still valuable people in those communities, and we should be leveraging them as well. I've said this to, I feel like, many patient groups, right? Because at the trial population, what we may need, you know, perhaps in the burden, you know, they're just too ill to be able to participate, or they've progressed so far, it's too burdensome, or aren't really able to follow the needed instructions to do the outcome assessments, or can they really have, you know, that MRI done, for example, and lay still. But there's still valuable information from those lived experiences, and I think we need to make sure that we're not just focused on that trial population and what that looks like, but really the whole community when we're looking at trying to get that information, because those are important folks too, and they want to contribute. I hear this all the time. I'm so bummed I couldn't get into that trial. Yeah, we missed the cutoff because my diagnosis time took so long. Very common, right? That we hear in rare disease. And I push back and say, but you can still help. You can still help in a way. And I hope people are using those folks as well. I think the other side where I'm seeing great things is the folks that are preparing for maybe potential future drug development. They have hope because they're seeing diseases or disorders that, you know, are kind of similar to theirs and to their progression, and they're preparing their communities. And that's a very different conversation, right? So now they're having to educate about what is a clinical trial, what's eligibility criteria, what is an outcome measure, what does it really take, how do you make these decisions? These aren't things that necessarily FDA is a part of, but we know these are conversations that the communities are having. And in the big picture of medical product development and drug development, this is kind of part of that spectrum, right? Like I said, there's multiple stakeholders, and so this may not be our stakeholder part, but there's other opportunities. And so I hope, and I appreciate the diversity of experience of these diseases we have here, because they all have individual unique challenges of how do you handle this. You know, your patients are the patients with PKD, AR-PKD, they're going to have to transplant is an option, but we know that is not always available because of the need for organs and stuff. So, you know, there's such a wide spectrum with a lot of informative information from these experiences. So I just want to remind people that, you know, every disease and disorder is going to have its own uniquenesses, but I do think there's our common shared experiences collectively that we can leverage from.
Great.
I have just one thing when we talk about the gaps too, and I'll go really fast. I'll go really fast. But one thing I found I've been amazed at, and I'm grateful if any of you are here, if I read a paper and I don't really get it, or there's something about it that I don't understand, or I think that really that policy doesn't make sense to me, I can contact the author and I get amazing help. And it's just incredible to me how, especially in academic circles, my education from people I've reached out to try to understand something better has been incredible. And I think it's a resource that a lot of people don't realize. There are a lot of generous people out there who help a lot of us, so thank you.
Great. Good point. Thank you, Kathy. Building on some of these comments, how do we evolve and adapt with this data? As new therapies and interventions and practices are identified and improved, there's an impact on disease progression. How do we continue to adapt with these changes?
I feel like it's a Pat question.
Yeah.
Because I think Pat knows what my answer is going to be.
Well, I think it's very hard, because as these medical tools are implemented, the natural history changes. And I think this is where working with Critical Path has been really helpful to us to think about, all right, how do we look at contemporary natural history? How do we collect real world data? What are these PROs looking like? So this is sort of the totality of evidence we want to look at because of the changing natural history of a disease. But I will say that it's in our disease, in many of these, you're just pushing out the trajectory of the disease, right? You're not reversing it, you're not magically curing it. So this is where patient engagement works very well, and this is where trials are adapted, and this is where I think in clinical trials, if we got to rule the world, everyone would be involved in clinical trials who's interested. And then there would be a subset analysis so that they could determine you know, what that drug was doing and how beneficial it was, and obviously the risk associated with it. But I think this is a reason for real-world evidence and a reason for these PROs and a reason to understand what the priorities are in these patients.
Yeah, I was going to say, so the big thing is we have to always reevaluate the natural history of the disease, whether we have a approved treatment or not, because standard of care has changed over time, right? And we've had lengthy, and why I said Pat's good at this answer is because we've actually had this conversation with PPMD for a couple of years now on the changing of the natural history, the use of steroids, how's that really impactful, and then now that there's approved therapies. And so it is really important to think about the data you're collecting in natural history, because that is important. That's important to everyone, not just us, right? It's important to a developer and really identifying the right population. What are the right things to be measuring? Can we pool all this data together and have some fancy advanced statistics that Klaus tells me about all the time with modeling and things like that? But it's really important, and folks need to be willing and open to changing the data collection of their natural history over time as things change, as treatments change, as modalities change. It's really important. So in the ALS space, their natural history is now changing, you know, because of the recent approvals, and we have to make sure we capture that, because now that's a new thing, and that's not part of standard of care. We have to account for that and a lot of things. So I think that's an important thing, is you need to evolve your data collection as not only treatments become available, or maybe a new medical device to help other things, or if just standard of care has shifted in a way, is really important.
Excellent. So that kind of begs this other question, which is there's only so many patients available for clinical trials. So how do we maximize our trial designs while balancing these unmet needs of rare disease patients and appropriate endpoint selection? Go ahead, you just chime in.
Well,
Sorry. I'm sorry, go.
So I think one attractive thing to consider are platform trials. Not always popular with industry, but you know, have some benefits. There's a tendency for a patient who's tried one experimental medicine to stay on it for a very long time, kind of regardless of the results, and be kind of taken out of circulation to be included in other trials, to get at the key point of your question. But in the setting of a platform trial, patients, you know, if the results aren't promising, can be switched to something else.
I think one thing that AFTD has done is really try to create spaces where we can have pre-competitive collaboration, because there are a lot of different clinical trialists in this space, and they're all facing essentially the same challenges when it comes to trial recruitment, hoping to get advice from people like Michelle on outcome assessments and novel clinical trial design. So we don't conduct research ourselves, but we have the ability to convene all of the stakeholders in one room and just talk through some of those challenges, including people with direct lived experience in a way that's clearly not theater. When you hear people with lived experience giving their insights on those things like clinical trial design and recruitment, they're really giving very concrete, I think, tangible advice to people conducting the trials. So I think the more of these kinds of conversations we could be having in a pre-competitive space, the quicker we'll be getting to effective treatments.
We had a few years ago 2 caregivers, one of whom I think is in this room, went through clinicaltrials.gov and counted up what we call seats on the bus, how many seats there are in ALS clinical trials. And the number was staggeringly low. I mean, I think 1,500 would be a very generous count. And this is a disease with, at any moment in time, there are 30,000-ish people living with ALS, with around 6,000 or 7,000 new diagnoses in the US every year. So there's a problem there, and that's one of the reasons that it makes expanded access so important to people with ALS. And expanded access, honestly, 3 years ago just wasn't happening. some people with ALS and caregivers got together and decided that we needed to put stars on trials, not on the science or the promise of the trial, but on the humaneness of the trial. So they came up with a rating system called, it's PACTD. So if you Google PACTD rating ALS, you'll find the PACD rating system. And they looked at things like open label extension. We had an awful lot of trials that if you were in a trial and you felt like it was working for you, you were out of luck at the end of the trial. And that just is cruel. We had very few trials offering EAP. We had long washout periods in some. So this covered all of those things that, minimize exposure to placebo if you can with a platform trial. Do some things like that. So they started putting stars, PACD ratings on trials. And to prove that it works, one company had a therapy that had 4 stars. And they came back to the team and said, how do we get 5? So that's proof that these things are important to companies, and that rating system, I think, has really had an influence on ALS trials.
I think if we look at the Children's Oncology Network and how that was put together, Children's Oncology Network has been fantastic in terms of childhood leukemia, where most of the children died and now most of them live, 90-some%. I think if we could do basket trials in rare diseases or platform studies or platform trials, we have had a Type C meeting with FDA, and I know developers are not very excited about that because they lose control, or they feel like they lose control. But this is the way to open it up to our community, to better understand what works and for whom that works and the degree to which it works. And I think in our current, we are lucky to have about 40 different biopharma companies creating or developing drugs, and that's really wonderful. But for, I think, 30 years, we've talked about a combination of therapies is going to be needed. But if we do these one by one by one and wait till an approval, and then another one and wait till an approval, combination therapies aren't going to be here for this generation of boys. And they really do deserve combination therapies. Because if most parents believe that if you get a little dystrophin on board, whether that's a microdystrophin or an antisense, that's sort of the holy grail, if you will, and you can build on it. But for the children that are not amenable, or the young men that aren't amenable to a gene therapy at this moment because we aren't treating, or the label doesn't read for non-ambulatory patients. In fact, the window of our first gene therapy approval is 4 to 5-year-olds. You can imagine if you're 5 years and 6 months, and the process takes so long. So we have to think differently and be able to look at a particular drug and then wash out and go to the next one, so that we begin to collect the evidence and then we deliver those combination therapies that we've been talking about for 30 years. So I think rare diseases needs and deserves these platform trials. I think it's the best way to treat that population. It is heartbreaking for these families to see a trial that's ambulatory patients, and your son just went off his feet, right? And think of those young men who are 20. Have we given up on them? Are we saying, like, you're too old, you're too involved, you can't? So we have to do things differently, and I completely agree that we need to do platform studies.
So I think this is where we encourage to our industry developers or drug developers, this is why we say come talk to us early and often. Because that is our opportunity to have these early conversations, because we understand that we have a limited population to work with, and we need to make sure we're maximizing and we're not wasting patients with what is the data that needs to be collected. And I'm hopeful in the next panel that is going to kind of give that perspective to hear, because I know there are some industry folks, I see them in the room, who may be able to speak to why it is important to come have those early conversations with us. And actually, I would speak on behalf of my other regulatory agencies. Talk to them early. I think you heard that in the last session. Last week, I was at another meeting where the chief medical officer from EMA was there, and he was like, come talk to us often too. Rare disease drug development's global, and I think all the regulatory agencies you want to engage with will say the same thing. Come talk to us early so we can really be able to think through a trial design, endpoint selection, the safety data that needs to be collected. I know we're talking a lot about efficacy, but safety is still a critical part. If there's manufacturing questions, particularly in the gene therapies, We know there's a lot of stuff they manufacture there, or small molecules and ASOs, there's manufacturing stuff. This is why we say come talk to us early so we can start thinking those things.
So we have about 10 minutes left. I will open it up for questions. And if I don't see, oh, I see people moving towards the mic, so I won't ask anything else in between.
Hi, good morning. My name is Jacosi. I'm a senior patient advocacy leader at Spark Therapeutics, and I just want to thank you all for your time. My question relates to hope. I heard hope referenced, and I think that I have worked in the rare disease space for a little over 10 years, and I've worked for multiple industry representatives. And many of those conversations internally where I am pushing that we engage earlier, they shake their head and say, we don't want to raise too much hope too early. And so I'd love to hear this panel speak to the importance of hope and the capability of communities to balance hope against the realities of drug development, and what you would want us to understand about the role that hope plays in all of this for you.
Go ahead. Anybody want to take that one?
I guess I'll just say for us, we've always had a little bit of a firewall, I think, between pharma and our community. We were a little nervous for whatever reason about sort of having industry at, say, our education conference, which is a family-facing conference. And this past year, we had a number of different companies come and speak about their clinical trials, and the reaction was overwhelmingly positive. People were so excited to see that there was investments in this rare disease. Even if the clinical trials they weren't eligible for, their families weren't eligible for, just seeing that there was movement in the space, seeing that there's things going on at the basic science level as well as clinical trials was really, I think, quite thrilling. We got, again, really, really positive feedback. And I think one thing that we're always working against is a little bit of the paternalism in kind of the healthcare system of being scared to give the diagnosis when there's nothing that can be done. And I think sort of communicating to healthcare providers as well that there are opportunities now, there are things that can be done, whether it's genetic counseling or finding spaces for people to meet with other families that are experiencing the same thing, even if it's not an approved therapeutic that is going to solve the problem right now. Seeing the momentum, at least for our community, has been really, really exciting for them.
I think the word hope, it's complicated. And I think it's really important that we keep it real. And that we see people all the time who give hope that's not realistic. And that, to me, is terribly wrong. At the same time, I think if we can keep things real, then go through the portfolios and such and talk about it, people can come up with a realistic and a real hope. But I hate it when I see people manipulated with hope that is not realistic. At the same time, I think that those of you who are working on therapies, keep it real, because it's really important that people learn about those things because they want to either benefit from them or be a part of them. So thank you.
I think hope is a tricky thing. And I think that it's really important to think about our language when we talk about this. When people, so the first time we heard about antisense oligonucleotides at an annual conference, the basic scientist got up and he flashed a picture of a 65-year-old playing tennis. And he said, that's where we're going. We have a progressive disease, you're born with it. You have muscle degeneration when you're born. That 65-year-old didn't have it. He was operating on a protein that was smaller than the normal protein, but certainly a very efficient and stable protein. So language matters, and no one could achieve that right now or then, which was 10 years ago. So I think hope is really captured in language, and I think developers need to be mindful that patients and families are listening to the words they use. And when you use a word like cure, it's everything we want and nothing we can have, because this child, in our case, is here and has some degeneration. I will also say to you that parents are very realistic hope is always present for another day. I don't think there's a time that they go to bed and wish for this disease to stop here. So they're not asking for magic, even though some of us would like magic wands. So they're hopeful for another day of stability. And I think keeping it real about this is what it will do, hopefully, this is what we're hoping to achieve, and always in mind of wherever you speak as developers, whether that's an investor call and you're saying, this is magic, we're listening to that. So language matters. Hope will be there as long as you're realistic, and patients and families want real, because that is their hope for the next day and the next promise. And they're hopeful because you're here, because you're developing something. They want to be seen, they want to be heard, and they want to belong to a community. And for them, that's hope, and that's what they need.
I think we have time for one more question.
That was a really good question that's hard to follow, so I'm going to lower expectations. Okay.
Lower the expectations.
My question is to Ed, but it borrows from what Pat was saying, which is this recruitment by function and basket trials. In the work that NORD is doing, are you looking across, because we all talk about the and Michelle, you referred to it, the heterogeneity of all these different rare diseases. But if we take a step back and look at the functionality, we may find that it's almost better to look at people by their profile versus the tag or the disease that they were given. Are you guys doing that? I don't know if with the RDCA-DAP work or with the work that you're doing, because that may be just as informative.
Well, let me see if I can understand your question. Is it the thought that some diseases may be biochemically similar and you could put them in a basket? Is that kind of what you were
Clinically, clinical manifestations may be similar, so that to the point that Pat was making about those basket trials, if you looked across and stopped saying, oh, they have this, 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 and this condition, you say, oh, they have this clinical series of manifestations. Maybe these therapies would be relevant for this patient profile versus this disease.
Right, so if you have patients who are clinically similar, I think you can learn from them as a group certain things like, is a particular assessment tool, reliable and repeatable and all the kinds of things that Michelle would like it to be around that clinical presentation. But if the underlying disease mechanism is different, then very likely it's going to require, different treatment. So I think there's times when you could look at the larger population and times when you could split it. And maybe the clearest example of that is that there are very many genetic diseases that have essentially the same presentation but are caused by mutations in different genes. And so you could learn a lot by looking at the larger population of all the patients who have that disease, but certain treatment approaches, some of which have been mentioned here today, like gene therapies and ASOs, are going to have to be gene specific, so then you're going to have to separate them out.
So I just want to tie on to that. So I'm hoping Ramona later will talk about this in her talk, because it's something I've been fascinated with since last year's RDCA-DAP meeting, and something I've actually been discussing for RDCA-DAP for the last couple of years, and it's actually in the rare epilepsies. So fingers crossed the task group's going to start soon, because I actually have that same kind of thought myself, right? Is, because of genetic advances, where groups of people might have had a different diagnosis, they're now, because of genetics, it's actually something else, right? But when I talk to families in the rare epilepsy space, they're all experiencing the same thing: developmental delay, cognition, speech, communication. I mean, the list goes on. And so I've been like, I heard Ramona talk last year on ontologies, and she'll talk later, and I'm so excited, because I was like, whoa, this is a place to start. Let's explore this, right? I don't think it's not going to solve all of our problems or questions, but can that information maybe help inform us on endpoint selection, biomarker selection, how we collect safety data? I actually do. So I am optimistically hopeful. And I'm using the word hope, so maybe I shouldn't. I'm optimistic.
You're allowed.
I am, okay.
Yeah, you're allowed.
I'm optimistic. I think it could help us. I think we have to explore it as an option. I don't think it's going to solve all our problems. I think the individual drug development questions we'll still have to tease out, right? But I do think, could that help us in other ways as we think about a study design? And I think when we envisioned RDCA-DAP 4 or 5 years ago, we thought about, could we do this concept of like a family approach for things? I think it helps when we don't have when we have rare diseases that don't have a lot of numbers, and if we can pull together to kind of help get some better understanding. So I'm optimistic. I'm hopeful next year we'll have some more things to say. But it is something I know that we've been thinking about, and I do think, to Ed's point, it's not going to solve all of our things, but it could help us. And we know that as science advances in the genetic side of diagnosis, we're going to be running into this more probably in other diseases and disorders where, okay, there was a deletion here, or a splice here, or something for that.
Excellent. So thank you to the panelists. It's been a fantastic conversation. I really appreciate all of the information you shared. And I'm going to hand it over to our next panel. Thank you so much, everyone.
Good talking to you. Yeah.
Okay, what a great discussion, as Tom said. So up next on the agenda, we are going to be talking about aggregated databases in rare diseases and how they can be used to inform drug development. We'll be looking at a case example from FAICD and RDCA-DAP. So as soon as we exchange mics here, we'll welcome our panel to the stage. So. Okay, great. So I'd like to welcome Alex Betournay. He's the Executive Director of RDCA-DAP, and he'll be moderating this panel.
All right, good morning, everyone. Going to start this next session with case examples of the work we're doing in Friedreich ataxia and talking about the FAICD and RDCA-DAP efforts for this disease. I'm just going to set the stage with a few slides, and then we go into a panel discussions with a Q&A. What's the big red? I want to see a green. A big green then. All right. So a few disclaimers that we're going to pretend to read for 5 seconds. There we are. Today, really I want to discuss the Friedreich ataxia databases that we have at CPAF, the FAICD, historically the first database for Friedreich at CPAF, and then the RDCA-DAP, discuss the role of data sharing and aggregation with our panelists, some of the solutions and impact for the disease that we are already having, including informing drug development for Friedreich ataxia. So just historically, and we have Jen Farmer here from the Friedreich Ataxia Research Alliance to discuss further this effort that really started a big database for Friedreich ataxia that we have at CPAF. So this effort is called the Friedreich Ataxia Integrated Clinical Database, or FAICD, that was started in 2018 with really an impetus to accelerate research and drug development for the disease, standardized multiple data sets that were already available at the time, including natural history studies and clinical trials, into CDISC, the regulatory grade standard for data. and also make that data publicly available through this database, internal database at CPAF, where approved researchers could access and download some of these patient-level data that we were able to aggregate over the years. That was really a work started upon FERA wanting to collaborate with CPAF, and it has been a collaboration ever since between CPAF and FERA, and then FERA and RDCA-DAP. So you can see here the entry portal that is still available on the CPAF website, where you can potentially request access to some of the data for Friedreich ataxia. This is the current database, so as you can see, it's pretty compelling. It's probably the biggest rare disease database that we have at CPAF after Duchenne muscular dystrophy. That includes a big natural history study conducted by FERA called the FA-COMS, multiple clinical trials and control arms for clinical trials from industry. Recently, that database is growing. So recently, EFACTS, the European Foundation for Friedreich ataxia, joined the FA-ICD team and also shared their data. It's a multi-site collection of data across European countries, which is under curation right now and will be available soon on RDCA-DAP. Additionally, FERA shared a new natural history study, this time for pediatric Friedreich ataxia, adding again to the database. There will also be an update of the FA-COMS natural history study that will come soon to add and increase the current data that we have for the FA-COMS study. Also recently, our team got access to an EHR synthetic data set that is under exploration right now, so we don't know yet the full potential of that data, but early exploration is showing potential access to medication information and lab tests that could be interested. So we're taking a current look at this data, which include over 1,000 records. Now, the RDCA-DAP platform, you will hear tons about it today. Really our platform for rare disease data aggregation at CPAF, platform dedicated to maximizing the utility of the data and the use of the data. Utility because we're really implementing, the team is implementing a specific format to maximize what you can do with the data, create innovation in the way you can discover data, analyze data, maximizing the use because there is a public interface that has been open for maybe close to 2 years now, where users can access the data and take a look at the metadata and then potentially make a request for patient-level data and analyze the data online. So we have some of the Friedreich ataxia data set available right now, including the aggregate, and some users are taking a look, including one of our panelist teams today, at the data on RDCA-DAP. So you can see here that is through the RDCA-DAP portal, just typing for Friedreich ataxia, you get a list of data sets, and in green the FA aggregate is available for request. And we are integrating, as I mentioned, the European data. The key angle here is that we needed to put that European data on RDCA-DAP because it's fully GDPR compliant and has been built so that we can also accommodate international data, which was really critical for us to be able to get that data shared to us to further the effort in Friedreich ataxia with that European natural history data. And you can see here, one of the key aspects is that there is a little more more security and privacy when you access data on RDCA-DAP. For the coder database, it was a download of the data that you can get to use on your computer, but on RDCA-DAP, it's very controlled. You cannot download the data at the moment unless there is a specific request for that by contributors. So it's providing security on secured workspace with tooling so that you can analyze the data directly, and we're trying to build tools that you will hear about today to facilitate the data analytical directly on the platform. So this is an output that really shows, that I really like that graph. It really shows the extent of the data that we have, because this is data for the Friedreich ataxia rating scale, and the black dots are individual patients through the database. So you can see the tremendous amount of information that we have on one of the critical endpoints that is used to track Friedreich ataxia patients. So, we are programmed to RDCA-DAP, and our goal is to leverage the data and build solutions to accelerate drug development. I'm not going to go into details, but one of the ongoing work that we are doing right now internally is to develop disease progression modeling for Friedreich's ataxia. We talked a bit about that in a session yesterday. We're trying to advance clinical trial simulation tool for the disease. That's the RDCA-DAP work. There is a tremendous amount of publications that came from the FAI-CD members, and we can send over publications. Typing David Lynch, Friedreich ataxia, or Christian Remy, to name 2 members of the FAI-CD, you see a lot of critical publications that were made throughout the years. On that note, I will introduce our panel today. So we have Jen Farmer, CEO for the Friedreich Ataxia Research Alliance, Shannon Rich from Rare Pharmaceutical, and we'll talk about the first approval, first drug approved for the disease that Rare had this year. And Shannon is Associate Director of Clinical Data Science at Rare Pharma. And Robert Chuck from OCP FDA. Robert is a clinical pharmacologist. I'll give you a chance to introduce yourself further, do a better job than me first. Jen, if you want to start, maybe tell us more about the efforts at FARA and really what was the original intent for you to come to CPAF, to create that database, and where you wanted this effort to go.
Sure. Thanks, Alex. Hi, everyone. I'm Jen Farmer, and FARA is 25 years old this year. We were started by patient families and scientists, and as an organization, really focused on advancing research in Friedreich's ataxia. Most of our programs are centered around, funding basic translational clinical science grants, as well as, what some of the previous panelists talked about in terms of looking at that pre-competitive space in drug development and trying to put the tools in place for the community so that we can accelerate and further inform the drug development process. And so the inspiration for creating the FAI-CD came from Jane Larkindale, who's sitting in the back of the room, who was working with me at FARA a while back. At the time, we had been collecting natural history data for a little over 10 years. And we also had several failed clinical trials. And one of our previous panelists, talked about the importance of patient contribution of data and how hard it is, for individuals participating in natural history studies and clinical trials to be contributing that data. And so we were really motivated to start this integrated database so that we were really leveraging every single contribution of patient data that was being put forward, whether it was natural history study or clinical trials. And we also knew we had to be smarter about how we were designing these trials, given the failures that we had had in the past. And so that was really, kind of those different things coming together simultaneously led us to the creation of the FAI-CD. And, we were really fortunate that a lot of the industry partners that we had worked with previously on those trials were really ready and willing to contribute the data from those trials to the database.
Thank you, Jen. And just to emphasize, it is a tremendous achievement to get so many studies, and it was actually pretty fast. So my day-to-day of trying to get data shared to our platform, it is quite an achievement to get that fast, and we're still building. Next, Shannon, if you want to maybe tell us more about your role at Reata, and then why it is important for industry to be able to access this aggregated database. And if you want to extend, we'll continue with you a little more to discuss specifically what you did on the FA data. But first, please tell us a bit more about your work.
Yeah, I'm Shannon Rich. I'm the Associate Director of Clinical Data Science at Rare Pharmaceuticals. I've been at Reata for 5 years, and I've had the opportunity to work on clinical development programs in Friedreich's ataxia, as well as other rare diseases. And I guess a little bit later I will share some more about my experience using the FAICD database. But first, to answer your question about the benefit of having an aggregated database, I think it's beneficial really throughout the drug development process. It can inform trial design, in terms of having that aggregated patient-level data that you can decide how you want to analyze. And so you can really answer very specific questions about a patient population that you're interested in. So getting information about disease progression and baseline characteristics, which can allow you to understand, the expected change from baseline and expected variability in a given endpoint, which can better inform the sample size of a trial. At the end stage, after the trial is completed, I think that having access to that data from other trials and natural history studies can allow you to really more deeply understand your own trial data, because you can examine your trial results compared to the other studies in the database.
Thank you, Shannon. Now, Robert, some question. Maybe tell us a bit more about your work within FDA, and also, maybe the angle from the regulator perspective and someone at OCP on being able to access this aggregated database, again, from the regulator perspective.
Thanks, Alex. And I think I inadvertently sent an older bio that's a little bit outdated. I'm currently the Deputy Director of the Division of Translational and Precision Medicine, which is still in the Office of Clinical Pharmacology at FDA. So I've been at the agency about 10 years, starting in the Genomics and Targeted Therapy group, which is a group that works on precision medicine approaches across different therapeutic areas. And a few years ago, we combined with a team working on rare disease drug development and really trying to use translational science approaches to advance drug development for rare diseases. So that's kind of our current goal, and one of our goals in trying to access the RDCA-DAP database. And really try to use it to figure out how we can come up with better approaches to develop drugs for these groups of patients. Why do I think it's important from a regulatory perspective? I think we're always, as others have said, we're working with a paucity of data in rare disease drug development. So we really need to take advantage of every piece of information we have about these patient populations in order to see if a drug is safe and effective for them. And that's where we can really use resources such as RDCA-DAP to combine data sets, look at biomarker development, design better clinical trials, and really make sure that we're doing the best we possibly can to give these drugs a chance to prove that they're safe and effective in the patient populations that they're being developed for.
Thank you, and apologies I scorched your title. Good job, Alex.
No, I think that was what I sent in.
All right. So yes, Shannon, maybe back to you. Can you tell us more about really the Reata story? So again, the first approval for the disease arrived this year, which is like an amazing achievement for a patient population with a terrible unmet need. So, Jen, a comment first?
Yeah, can I just because of the prior session and the importance of patient engagement, and I loved the discussion about disruptors in the field and family members who become citizen scientists. That's actually where this story starts. And so I thought it was important to maybe just give you a little bit of that backdrop. There's a mom whose son was diagnosed with Friedreich's ataxia, and she was up every night reading every PubMed article she could find to understand more about the disease. And there had been a few publications looking at how the protein deficiency in Friedreich's ataxia leads to downstream metabolic and mitochondrial consequences. And she comes across this NRF2 pathway paper that has been published by some of the academic scientists in the community. And her husband was a chemist and supplying API to biotech and pharmaceutical companies. And so the next morning she says to him, NRF2, is that a druggable target? And he happened to know the answer and said, yeah. And she said, do you know any companies working on drugs that target NRF2, and it turns out he was working with Reata and helping supply them with API for some of their other products. And she picked up the phone and called Farah and talked to Ron Bartek and said, listen, you know, this could be just some late night mom Googling and reading papers, but I just feel I have to share this with you. And Ron, of course, was all in and interested in what she had to say, and followed up a quick introduction by her husband to Reata with Ron, myself, and one of our lead clinician scientists, Dave Lynch, flying to Dallas and like knocking on the door and saying, hey, let us in. We want to talk to you. We want to tell you about Friedreich's ataxia. And at the time, Reata was not at all interested in developing drugs for neurologic diseases. They were focused on kidney disease. And so we kind of really surprised them. This was kind of out of left field. But, you know, I just wanted to share that as the backdrop with our prior session about, you know, really engaging your community, the disruptors, and our role as advocacy organizations in facilitating that process and kind of bringing people together. That's where this story starts. And I'll turn it over to Shannon to also, you know, talk about the clinical trial design and the work that we've done together since then. Thank you.
Yeah, thank you so much. Yeah, so our trial in Friedreich's ataxia is called the MOXIE study. It was conducted in 3 different parts. So part 1 was a dose-ranging, placebo-controlled trial, which patients were dosed for 12 weeks. Part 2 was conducted in separate patients. It was a placebo-controlled trial as well as our pivotal study, and patients were dosed for 48 weeks. And then eligible patients from both the Part 1 and Part 2 studies could enroll in an open-label extension phase. And so the extension began enrolling, I believe, in late 2018 and is still ongoing. And so we've collected many years of safety data as well as some efficacy data. And so in our Part 2 pivotal study, what we saw or what we observed was a significant improvement with our drug relative to placebo in the primary endpoint, which was change from baseline at week 48 in MFARS, which is the Modified Friedreich's Ataxia Rating Scale. And so with these positive results, we wanted to use the long-term efficacy data, MFARS, that we had collected in our extension phase to support these pivotal trial results. However, because it was an open label extension, we didn't have a control arm to use to kind of best interpret the extension results. So we requested the FAICD database, which had the FACOMS data, the natural history study, along with some other trial data, so that we could actually use that data to provide an external control to better understand our extension results. And so I guess more specifically, we used the natural history data and performed a post hoc propensity match analysis. With this technique, patients from our extension study were matched 1-to-1 with patients from the FACOMS study. And the matching was done based on sex and then other prognostic factors for disease progression that were available in both of the studies. And so this was baseline MFARS score, baseline gait score, which is a question within the MFARS, age, and then age of onset. And so we came to these factors with discussions from Farah and Dave Lynch, who's been mentioned, and statisticians at Farah. And then I guess one other really important thing to mention is that within the FAICD database, you know, there are different studies, but the FACOMS study was really the one that was beneficial to us because of its really large size. So it made matching 1-to-1 feasible. The FACOMS study is also the only one that collected the MFARS data over many years. And so obviously we needed, you know, long-term data collection so that we could analyze our long-term results. They also were very robust in collection of demographic and baseline characteristic data, which is what we needed to be able to match those patients and really generate a comparable external control group. And so, all these factors really made the FA-COMS data within the FAICD database the one we needed. And so after we matched the patients, we analyzed change from baseline in mFARS over 3 years in our extension patients and in the patients that they matched to within the FA-COMS data set. And we observed an improvement in mFARS in extension patients after 3 years of treatment compared to the untreated matched FA-COMS patients. And so I guess just in summary, having the FAICD database and the FA-COMS data specifically really was beneficial in allowing us to interpret our open label extension results and in providing some support to our pivotal Part 2 study results.
Thank you, Shannon. Trying to see where to go next because I have 2 angles here. I'm going to start with the RDCA-DAP piece. So as I mentioned, we transferred some of the data that is already available. One of the reasons for that is as we want to continue the effort, there's a need for globalization in Rare. Really, if we want to get somewhere and push things forward, we really need our efforts to be international. And so RDCA-DAP was built in part for that reason in Europe to be able to enable that international aggregation of data. So some of that data is available. Robert, some members of your team are taking a look at the Friedreich ataxia data. Maybe can you give us a few highlights on, I know it's early, but some of the interest that maybe you see from your team's perspective on using the RDCA-DAP as a platform to take a look at rare disease data?
Yeah, certainly. So I think the case example is a great one of one potential use of this platform. I mean, it's highly impactful that they're using it for, actively using it for part of the regulatory approval process. That's very impressive, especially as early on as it is in the use of the database. So I'll also just say in full transparency, from the FDA's perspective, I was not involved in this review in any way, shape, or form. So kudos both to Riata and to the review team. I'm simply commenting here as a bit of a spectator. But I do see many other potential uses for this type of platform beyond just that direct application in the drug approval process. Our group is looking at it as more of a research tool. We're looking at different ways that we can use innovative approaches to advance drug development. One of those is better understanding the disease itself, and that's one big advantage I see of letting everybody be able to access this data. You have many different minds and many different people who can think about things in different ways and come up with ways to better understand a disease, and that can help us with things like biomarker development, pharmacodynamic biomarker development, looking at correlation between baseline factors and prognosis or outcomes in patients, really just helping us better understand what because we have such small numbers of patients, what we can expect to see in a clinical trial, and that again helps with that clinical trial design, helps us have additional endpoints, come up with ways to have confirmatory evidence of effectiveness. I think that's how the database was used in this case is in regulatory speak, it's a way to bolster our substantial evidence package is to have a piece of confirmatory evidence. One way to do that would be a natural history study, as was done in this case compared to part of the trial. Other ways would be to have like pharmacodynamic biomarker effects can be used as confirmatory evidence, but we have to understand those biomarkers and how they typically behave in that disease. So by looking longitudinally across databases like this that have biomarker information, we can better understand whether something might be a good pharmacodynamic biomarker, whether that could be a piece of evidence we could use in a future submission. So we're really taking more of a research-based approach. It's still pretty early on, so we're kind of figuring out what's in the database, what we can use, what questions we can ask, but are looking at kind of being able to better understand the disease as a whole and how the next drug development program might look like.
Great, tremendous. And like speaking on behalf of the team too, we really appreciate all users that are currently using RDCA-DAP getting back to us with feedback. We all strive to make the platform better, provide new tools and innovation so that we can really continue to improve what we already have, which is already great. So Jen, moving on now, I'm sure there were lots of tears of joy in the community, and then boom, the aftermath. This is not a cure for FA, and after years and years of work, it was a big relief, but now it's okay, moving on. where do we go next? So maybe can you comment on what you see as ongoing challenges and maybe opportunities to continue the efforts for your community?
Yeah, absolutely. There's the need, I think Pat said it beautifully. We've got to continue collecting this natural history data, right? We need to understand how, as treatments get approved, the natural history changes. We need to get new tools in place for the next generation of clinical trials. We need novel trial designs. And so we're actually kind of doubling down now on this initial investment and trying to grow it even further. And the European natural history study that Alex mentioned, we're actually combining that into a single natural history study, FA-COMS and EFACS coming together into a unified study, so that we can have more than 3,000 individuals with FA globally engaged in natural history studies, continuing to collect this data. Being able to support post-market registry studies, that's going to be really important to us as well. The conversations that have been happening here around pediatrics is also a high priority for us. The drug is approved currently in individuals 16 and older, and we need to start trials in individuals with FA younger and younger, and understand how best to do that. So Pat came over to me and said, "How are you doing?" Because you thought it was hard to get to the approval. You get to the approval, and then it just really takes off, and then things get really crazy, and there's a whole lot more work to do. So these kinds of efforts are ones that are not just a 2 or 3 year investment. We started FA-COMS in 2003, and it is 20 years in now, and it has to be something that we can figure out how to sustain and grow so that we can continue to evolve our clinical trials and bring new treatments forward, and that's what we're thinking about.
Great. Maybe I'll relay the same question, Robert, if you want to start, maybe commenting now. We have a new baseline therapy. What's the impact, and how should we move forward with the future clinical trials?
Yeah, well, one thing I'll say is that often, once you have an effective therapy and you have patients on an effective background, it can become harder to actually demonstrate efficacy for the next drug unless they happen to be synergistic. So, thinking about, that means we need to do a better job with clinical trial design and all the things that we've been talking about. In terms of understanding the disease, designing the best trial, having the most effective endpoints that are going to be able to demonstrate an effect for the drug. And again, I'll kind of come back to what I've been saying. I think using it as a research tool to understand how we can improve all of those elements, improve that clinical trial design, discover biomarkers that can be used as pharmacodynamics, and able to show an effect there. And really just be able to prepare for what the trial is going to look like, because as everybody said, the environment changes once that drug is on board, and the natural history changes, and all the things that we knew aren't necessarily true moving forward. So, that's why it becomes even more important to understand the disease as well as we can.
Thanks. Jen, maybe a comment on that? I'm sure it's part of your consideration right now, continuing the data collection, like the baseline therapy.
Absolutely, doing all of that, and continuing to understand, how to develop better endpoints and biomarkers. At the same time, I think Michelle said it. You need to continue to evolve these processes, and so. At the same time, you want to keep collecting a lot of the same data so that you can compare over time, but you've got to keep iterating, and you have to keep innovating that for new endpoints and biomarkers. And that is something that I think we've done pretty well in our natural history study in terms of evaluating potential endpoints and biomarkers in small pilot studies, and thinking about looking at that data and deciding, okay, which ones do we bring in now that are part of the core set of assessments, continuing to look at the data we're capturing, getting rid of things that are not sensitive, that don't seem to be working well, and getting better and better tools.
Yeah, great. Yeah, and something like I didn't present, but really the FERA scientists did a tremendous job taking an overall look at 17 subunits of the scale. Yeah. Is that 17? Yes, 17 subunits within the scale across the database. All these subunits are tracking patients over time, which subunit might work better for which category of patient at which stage. That's really a lot of effort, and it speaks also to the, how great it is to have a collaborative environment as we're building our trial simulation tool, the first core, as we paused the work for a few months. As we restarted, getting the feedback from IFACT and FERA on the effort, telling us, well, we don't really care about this is where you should focus on improving your models because this is where we want to intervene. Seems obvious, but there's also a lot of conversation of, patients getting from ambulatory to non-ambulatory, the transition phase, how can we better find means to track and to measure that. There's, it's part of the challenges that we need to, and why we need to continue the work.
Maybe one other thing I'll say that's been, I think, a really good outcome of the FAICD and RDCA-DAP. FA is a small research community. It's relatively small. And we have one statistician that we work with as part of FA Comms. And being able to make these data sets available to the broader community, we've had more than 20, 30, I lost track, but I mean, we've shared this data with a lot of different companies and academic researchers and with the CPath team. And so we now have more and more people looking at this data, thinking about different ways to apply different statistical approaches to understand the data better, to help us with disease modeling, understanding the placebo effect that occurs in FA trials, and different approaches to clinical trial design for different therapeutic products that are in development. That's also been, I think, another important outcome, and one that we want to continue to facilitate is, having more and more people help us think about what ways we can leverage this data set for new trials and understanding the disease better, and it's been really important for that as well.
Okay, maybe a final comment from Shannon on what's next for READA. Maybe what was missing, what you would want to see as the database continue to increase.
Yeah, I think as Jen mentioned, the drug is approved in patients 16 and older in the U.S. And so obviously, there's a focus, to expand in the rest of the world, but also in children. And I guess just thinking about, kind of what's missing, or obviously, and you've been mentioning it as well, just continuing to collect more data. And you're doing it with EFACTS, but there are some differences in endpoints, and so getting that data that has more different endpoints will be more useful to other sponsors as well. I know just one challenge we faced while using the database was really just we were interested in concomitant medications, and it was collected in both studies, but there was some differences in the information that was collected, and we didn't know what coding dictionary was used, and so it made it challenging to really summarize those results across the two studies. We could get some useful information, but it was just hard to really directly compare them.
Mm-hmm. Great, thank you. And I think the HR piece, might be something we need to continue to discuss. We talked in the past about maybe the lack of information on cardiac data. There's really still a lot to do. So I will stop there and start the Q&A. Maybe just take a minute to acknowledge the moms, and your story was a great listen to your mom story. And we had a group of data scientists and moms of rare disease patients yesterday. So thank you to the moms. And let's start the Q&A. No, Cecile, you're second. There's someone behind you.
Well, she can go because I was
French
actually going to defer to the woman who I jumped in front of last time. So go ahead.
French culture, we have. Sorry about that.
No, I really want to thank the panel and this session about RDCA-DAP. So as I mentioned yesterday, I'm leading activities for CPAS in Europe, and I just want to really congratulate everybody, because everybody in Europe is so jealous about RDCA-DAP. Like, it's really a game changer, and it's really perceived with a lot of positivity and a lot of discussion about how to engage best and make it interoperable to accelerate really the development of global data set, to enrich data for disease modeling and DDTs. And I do hope that we can help you advance that project in a very efficient and timely way. So congratulations and thank you.
Now I have to come back with a question after all that praise. So, Shannon, you mentioned something that caught my attention about capturing the meds. And I'm wondering, actually, for the natural history data that you've been collecting, if you've thought about those factors beyond the clinical manifestations. A very good example of that is Erbitux, where they noticed some safety issues with patients who lived in North Carolina and Tennessee that was related to, eating barbecue, as we all do as Southerners. But like, that kind of information actually does guide the responsiveness to a therapy, but it's not something you'd capture in a clinical trial. So I'm wondering if you guys are thinking about that, or even for RDCA-DAP.
So Briata and Farah are working together to launch a post-market study where we can capture more of that real-world evidence and longer-term safety in a broader population, and leveraging, our Farah's clinical research network that does the natural history study to implement that post-market study. So I think we are thinking about that, where, we know that the number of people who've been able to access the medication through the trials is relatively small. It's about 170 or so people, and now, expanding that out to 4,000 individuals in the US, or there will be things to continue to learn, and thinking about how best to capture that, yeah.
All right, well, thanks everyone. Thanks to all the three of you for joining. It's always a treat to do a panel with Alex, I know. But thank you very much. We're heading into a 15-minute break, and the next session will start at 11.
Hi, everyone. If you guys can start making your way back in, we're going to get started in just a minute.
We can grab somebody else from the audience if you want here, too. Yeah. We got 75 minutes. This is a big game show. So feel free to elaborate. Thank you, everyone. We're going to start this session. We have an esteemed panel here with us today. Everybody come on and get seated. So this panel is regulatory pathway and applications in drug development for rare disease and case studies. So the purpose for this panel I alluded to yesterday. There's been a lot of recent approvals that, they disseminate into the broader community. There's, misconceptions sometimes about the totality of evidence that get discussed in the broader community. So what better way to address this was to assemble a group of people that could speak to different elements of, recent approvals. Obviously, there are some individuals that can't speak directly to their product, but they can talk through the steps on this panel. But what we wanted to do is address those so that people would get an opportunity to see, and hear examples of the, the dialogues and conversations, but also the levels of evidence that are needed to be done. So I'm going to start this panel with continuing the conversation we had regarding Riata, and Shannon, if you want to interject anything additional after you hear comments from the agency. We have Emily here, who actually did the review and can provide some background experience, and we'll come back and we'll interview people And after this, I just wanted to start that for continuation, and we'll go down the row.
Sure, thanks. So for those that don't know me, I'm Emily Freilich. I'm the director of the Division of Neurology 1 at Cedar. And this is a good continuation since we just heard a lot about omaveloxolone and Reata. And I think Dr. Baracchio also mentioned yesterday some of the considerations that went into determination of substantial evidence of effectiveness for that application. As you heard, we have to determine substantial evidence, and typically it can come from 2 studies, a single study, or a single study plus confirmatory evidence. And as we also heard, Reata, omaveloxolone did have a positive, adequate, and well-controlled study that won on its primary pre-specified endpoint. But it had some limitations as well. It was a small study. It did not have positive findings on the secondary endpoints. And so it wasn't persuasive enough to stand alone as a single study. And so we really were considering what could serve as confirmatory evidence to support what appeared to be a positive treatment effect in the single pivotal study. And they had presented also some mechanistic data that showed that the drug seemed to be doing what it was purported to do. So some biologic plausibility that the drug was having its effect on NRF2 activation. And the natural history study, which we heard a lot about in the last session, but the benefits of that study, or the positives that allowed us to really rely on it, were what we heard a lot about was the natural history database, right? This FACOMS, where we had individual patient-level data and from a large number of patients that had systematically collected the same mFARS that was the endpoint in the primary, in the open label extension for the pivotal study. And so the other thing that was also compelling was that a lot of the centers were the same, and so we felt confident that the data was collected in a similar fashion. I think there was overlap on a lot of the centers between the natural history study and the open label extension study. And so looking at that data and seeing that you had several years of progression in the natural history data and could compare it to the 3 years of open label extension data on the same endpoint on the mFARS, and we're able to do the propensity matching because they had collected demographic data for baseline characteristics to be able to match on the important prognostic variables. That was really what led us to be able to rely on the natural history study for confirmatory evidence. Now, there are always limitations to external comparisons, and this was not immune to that. It was not pre-specified. It was an exploratory analysis, but it was able to support the primary endpoint, and that was really how we were able to rely on it. It would not have been able to support efficacy of the drug in and of itself, as many open label external controls studies cannot do, although in rare disease that is an option sometimes to do an externally controlled trial, and FDA does have an external controls guidance that gives you kind of the considerations and limitations of using external controls.
Do you want to comment any more about that and other applications that you've reviewed and use of real-world data? I mean, any examples that you saw that were really effective or ones that might be more challenging? And you can keep it more generic and not throw somebody under a bus, but we'd like to hear more.
Yeah, I think one thing to consider, and this is not necessarily how we're thinking about it today, but the agency has been using real-world data for many years to look at safety, right? Post-marketing, we look at real-world safety data all the time to update labels, figure out when there needs to be a warning. And so we're not strangers to real-world data. There's also been a recent example in the Division of Neurology 2, where they were able to use real-world data to update the dosing regimen because there was safety data about the safety of that dosing regimen for an epilepsy drug. So I think real-world data use is out there. When we get to efficacy, it's a little harder because of the limitations But I think if you can pre-specify to the best of your ability what you're going to be looking at in the natural history study, systematically collect the data, in a similar fashion at a similar time points that you're going to do in your controlled study, and having that patient level data. Those are some of the key things to consider. And you can't control for all the confounding and bias, but the most you can do to limit that bias, the more successful you may be in having an externally controlled study.
We'll just continue on with introductions. So back, I just wanted to not lose that thought, and since we had the break, it would still remain in people's short-term memory. Steph, do you want to go ahead and
Yeah, I'd be happy to. Hi, everyone. It's a pleasure to be here. My name's Steph Fredette, and I work at Biogen. I oversee the clinical development aspects of our neuromuscular portfolio and led the development of the tofersen program for SOD1 ALS. And then my new favorite role that I just took on is a co-chair of the AMP-ALS initiative that you heard a lot about yesterday. So it's an honor to be here, and I look forward to the discussion.
Hi, I'm Dawn Phillips, Senior Director of Clinical Outcomes Research at Regenix Bio, and I'm excited to be here and to have this opportunity to share how we do endpoint modeling within some diseases I work in. I focus on rare. We have 4 rare disease programs, and I work across them.
Hi, I'm Allison Martin. I am a director on the regulatory science and policy team for North America at Sanofi. I span the different TA areas, but focus pretty heavily on patient-focused drug development, rare disease, and real-world evidence.
Hi, I'm Jessie Hanrahan. I'm the Chief Regulatory Officer at Solid Biosciences, which is an AAV gene therapy company dedicated to the development of neurologic and cardiac therapies for patients, including Duchenne muscular dystrophy and Friedreich's ataxia. Prior to my time at Solid, I worked at Bluebird Bio, where I oversaw the global regulatory strategy for their severe genetic disease programs, which included Zynteglo and Skysona, which were recently approved here in the United States. And prior to that, I worked at Genzyme across MS and oncology and rare disease.
Hello, my name is Larissa Loptila, and I'm the Associate Director in the Division of Clinical Evaluation and General Medicine in the Office of Tissues, in the former Office of Tissues and Advanced Therapies, but currently Office of Therapeutic Products in the Center for Biologics Evaluation and Research. And many of you probably know that our office oversees, although we changed the name, we oversee the same categories of products, and they include cellular therapies, gene therapies, tissue-engineered products, plasma-derived therapeutics, xenotransplantation products, and a number of other biologics. And most of our product development programs that we see are, in fact, for rare diseases. And I also look forward to the discussion.
Hi, my name is Mark Rudd. I work in Global Regulatory Strategy for Biogen. I oversee our neuromuscular disease franchise. So Steph Fredette and I work very, very closely. I'm her regulatory to her clin dev. We do get on very well. We just decided to sit a few.
She told me to sit you over there.
We work together.
She told you to separate you two.
The biggest privilege I've had over the past 18 months has been to lead the Qalsody new drug application and in the advisory committee meeting, which Steph moderated and presented on. So we have a lot of experience in rare diseases across AbbVie, Celgene, and GSK previously. And I said most recently, the one we'll probably talk about a little bit more about today is Qalsody, the accelerated approval in SOD1 ALS. And then we'll be friends again, right?
Okay, given that, Steph, I mean, there was a lot of excitement after the advisory committee and the subsequent approval of tofersen. Could you start telling the story and sort of building the level of evidence, and then we maybe can have FDA react to the readout, I guess, from the discussion.
Absolutely. I'm not usually organized enough to take notes, but Mark reminded me that brevity is not my strength when talking about tofersen, so I figured I would just write a few things down. But it's been an incredible journey, and one that started far before I was working on the program. It's in 1993, SOD1 was the first gene that was identified to be relevant in the pathophysiology of ALS. And it's an ultra-rare subset of the population it affects. I should say there are approximately 300 people living with symptomatic SOD1 ALS in the US right now. And 30 years later, in April, we had the first targeted therapy for the treatment of SOD1 ALS approved with tofersen. It received accelerated approval based on reductions in neurofilament light that were found to be reasonably likely to predict clinical benefit. And just very briefly, tofersen is an antisense oligonucleotide, and it's designed to bind and degrade SOD1 protein. It's trying to reduce the protein and the accumulation of that protein to preserve sort of the function of motor neurons. But I'll jump to the phase 3 study because I think there's so much we can learn. We knew that neurofilament was important. It's a marker of motor neuron health of the active neurodegenerative process, and so we were measuring it as a key secondary endpoint. But it was not our intent to pursue accelerated approval when we designed VALOR. We were hoping to demonstrate substantial evidence of effectiveness on a clinical outcome measure, the gold standard endpoint, ALSFRS-R, that I could talk about for hours if anyone wants to after this, in ALS trials. And so off we went and we designed a 6-month trial with an enriched primary analysis population, which we thought was the right thing to do at the time. And we had, unfortunately, an open label extension that I'll talk more about. And we designed the study based on the very limited natural history data that we had available to us at that time. And come to find out, we vastly underestimated the size of the study that we needed, and that's been an important learning as well. Not only is there limited natural history data, but it's also not consolidated, not collected in a consistent manner, just again emphasizing the importance of everything that the Critical Path Institute and NIH and others are trying to do. So in August of 2021, I saw the primary analysis, and I don't think you can get a p-value worse than what I saw that day. I thought it was a joke. I actually did. I thought my statisticians, some of you may have heard me say this before, were playing a joke. There were many tears, and then the next day I saw the biomarker data. I saw the reductions in SOD protein, and I saw the neurofilament data. And at the same time, I got transcripts from these qualitative exit interviews that Dr. Campbell actually recommended we collect in the VALOR study when we were having conversations about the phase 3 design. We didn't include them as quickly as I wish we did. We only had a handful of these exit interviews, but I was reading them, and I remember distinctly having tears streaming down my face because I was lining up the biomarker data and the exit interviews. And these participants were asked to weigh in on how has the disease impacted their life and how has that changed since they started participating in the trial. And they were so eloquent and so specific. A couple months in, nothing changed. But 2-3 months later is when I started to feel different than when I started. And looking again at the time course of the biomarker effect, I called my statisticians and I said, my goodness, it's working, and the trial design is all wrong. And so we had a long journey of being humble and really just trying to learn from the data, learn from conversations with regulators and the clinical community. We, a couple months later, had data from our open label extension looking at integrated analyses, early start versus delayed start analyses, so highlighting the importance of maintaining the blind from your parent study when you're conducting open label extension studies so that you're preserving the integrity of data collected in that fashion. And Mark can share more about where this bundle of fun that I handed him on the regulatory side to try to navigate. But ultimately, to jump to the sort of punchline, it was the totality of evidence, the pathophysiology of SOD1 ALS being relatively well understood, the mechanism of tofersen being understood, and really importantly, our understanding of what neurofilament is, what it's telling us, and the massive amount of work that the community had done to progress our understanding of neurofilament in ALS and identifying it as a prognostic marker of disease progression and survival that led us to ultimately having a successful advisory committee meeting and accelerated approval. The benefit of tofersen will hopefully be confirmed through the ATLAS study, which is the first interventional study, as far as I know, in pre-symptomatic gene mutation carriers. And as my friends at End the Legacy always remind me and others, is it's not a different population, it's an earlier part of the disease spectrum that we're evaluating, and likely the part of the disease spectrum that we need to be intervening in to really have a more profound impact on the disease. And so we're hopeful there. And the last thing I'll say is that while hitting the primary endpoint would have made life much easier, I really don't think that we would have learned as much as a community, certainly I wouldn't have, if that had happened. I feel like I've heard someone say that we broke the system with tofersen and really forced conversations that are happening now about what is the proper trial design, the importance of extension studies, the importance of following the science, the importance of collecting objective measures and all that. And I'm happy to expand on any of those points.
Yeah, you know, one of the things I noticed in your conversation is the amount of discussions. You didn't highlight as much the NFL parts of that, and that seems to be the most buzz, you know, people talk about. So for both of you, like, how did you prep for this, you know, when you're talking your regulatory strategy about inclusion of NFL? You know, what homework did you need to do early? You know, what types of preparatory things for your arguments did you think things through? I mean, any of that would be you know, that you can share would be greatly appreciated.
Yeah, I can start, Mark, with the sort of why did we include it as a secondary endpoint. I mean, I would say I spent a good amount of time working in the spinal muscular atrophy world prior to coming to ALS. And so I had a pretty profound appreciation for the importance of measuring neurofilament and what it could be telling us about the disease. But again, it's the work that the community had done to characterize it as a prognostic biomarker. So we were confident that it was important, and it was actually our second key secondary endpoint, which is a bit unusual in the context of a clinical trial. So it was part of our hierarchical testing and such. And so that part of it was pretty clear and didn't require a lot of conversation. We needed to be using the right assay and making sure that we were assessing in the right way. But then ultimately preparation for the advisory committee meeting, I can have Mark weigh in on sort of what some of the fun that went into that, and of course discussions with the agency.
No, I'd say the most important part for the team was that kind of evolution from when the phase 3 study really read out. So everybody in this room knows that drug development is not linear, and it's certainly not for rare diseases. But most importantly, post-study readout, we had to really work on how we were defining substantial evidence of effectiveness. How could we then best position the NDA for the agency to begin their review, as well as hopefully lead it to a successful outcome. So when we first came to the agency, the first discussion was around the feasibility of this data set in its totality of approach. Collin, as you mentioned in some of your introductory comments, when you had strong target engagement, you had substantial tofersen-driven reductions in the biomarker, you had clinical trends that were numerical, in an ultra-rare disease setting. And I think we had a very important discussion with the agency early that stated, you know, in support of a traditional approval, one must show substantial evidence of effectiveness on a clinical endpoint or a verified surrogate endpoint, which we did not have, as Steph discussed. And then really, as we were able to take the agency's feedback and really go back and look at our evolving data set, evolving because we also got to look at the open label extension data that really enabled us, with some statistical limitations, to look at the patients at 52 weeks. And I think we saw some really interesting data in there. You saw the delayed start patients, those who received placebo initially, have the same target engagement and the same similar reductions in neurofilament, and you could start to see the emergence of and the separation in the clinical data. So really, that then lent us to think, well, where are we getting our substantial evidence of effectiveness from? And it was very, very clear that it was on the biomarker, right? So when you see those substantial evidence of those substantial reductions in neurofilament, we then had all of our work cut out for us to then show that, in support of an accelerated approval, that those are then reasonably likely to predict clinical benefits. So there's an extensive research group within our company who were looking to really model that so that we could successfully position the NDA for the FDA to begin the review as a potential accelerated approval. But of course, we're doing this whilst we're building the NDA, right? Whilst we're writing all of the documents and all of the summary documents and having a lot of internal discussion. And then we really came to the agency at our pre-NDA meeting, right? If those of you who've been involved in this, it's often a little bit more tactical. And that's where we asked the most strategically important question of all of it, which was, based on what we can share with the agency at this early part, could those tofersen-driven reductions support an accelerated approval? And that's a tough ask for the FDA as well, because they've got a limited insight to this data. And I think having the benefit of the open label extension that was well controlled was certainly helpful in that, to have that time course. But then really all the fun was right ahead of us. And because, I really credit Dr. Frelich and Dr. Baracchio and especially the ClinPharm team at FDA, who were doing their own assessment on all of this data, right, to understand are the tofersen-driven reductions in neurofilament really reasonably likely to predict clinical benefit? And so we're all building this at the same time, really, and obviously responding to the agency's information requests and under priority review as well, right, which is a tough ask on the agency side as well as ours, while it's also then preparing for the advisory committee meeting. One of the other parts of this program that I think was really important that Steph and I had spoken about internally at length was we knew the advisory committee meeting, we knew we would have an advisory committee meeting, the PCNS committee meeting, at some stage. But we always felt it was probably the most important day for the program up until it became approved, because to really understand the tofersen story and the synopsis that Steph provided, you kind of need to see it all as an NDA. And we really felt that the advisory committee meeting, whilst I prepared for them before in oncology, we kind of dreaded them a little bit because they're kind of scary, right? There's a lot riding on that one big day and it's public. And this one we were really looking forward to because it was a chance to tell the learnings from the study, of how we would design the study differently, right? To be really transparent about the data set we had, how we believed we could show substantial evidence of effectiveness, and to hear from FDA and obviously the committee members themselves. So I think throughout that whole process, there's a lot of learning, a lot of internal debate, a good team that can challenge each other. Steph and I, that was mainly our night shift pretty much. When there's a day shift was operationally getting through the programs, and our night shift was outcome strategy and working through the different data sets. But a remarkable time and a remarkable way to really try to optimize as much of the regulatory toolkit as is available to us.
Joe, you add, or do you have anything you want to add?
I can add a little bit. I think that, and again, Teresa talked about this yesterday as well in the opening session, but it's exciting to have a biomarker be reasonably likely to predict clinical benefit in patients with ALS, but there are caveats, as we talked about, as to why this was possible for this population. And I think the review, shed light on everything that Stephanie mentioned. This is a targeted therapy for a mutation. We had evidence of target engagement of that, ASO on its target, and then the plausibility that the neurofilament light is a downstream effect of reduction in SOD1. And that, along with the data, which again, when we come back to substantial evidence of effectiveness, we had a robust finding on NFL that was persuasive, and the confirmatory evidence was the reduction in SOD1 showing the target engagement. And that together was enough to kind of get it to the finish line for accelerated approval. I think neurofilament light, as Dr. Baraki said yesterday, is not ready for prime time. It's not going to solve all of our problems, but it is a really compelling biomarker, and it can tell us a lot about disease. I think the more natural history data that we have on NFL in each disease state would open the door for discussions on when it could be helpful. As was mentioned, the data that has came out in the few years since the tofersen trials began and when it was approved really kind of changed the landscape and our understanding of NFL a lot. And our team, the ClinPharm team, really dug into that literature and how NFL could be used prognostically, and really helped us come to the determination that it was reasonably likely to predict clinical benefit.
And I think this, I would just say, and put a bit of a plug, I think at CPATH, one of the things this did was it put up a bit of a flare because of the conversations that have been brewing in other disease states around NFL. So we're discussing, amongst even our liposomal storage diseases, everything to ALS, is just to look at, a deeper dive of NFL and what we can, and under specific contexts of use, explore. So kudos to that. Allison, I wanted to touch base with you next. And, obviously you've been involved in some recent approval processes. And I wondered if you can share some of your experiences and things that you can say that are within the public domain, you know, and see your action with agency or things, the pearls that you might talk about for building your totality of evidence and
Sure. Yeah, thank you. And thanks for putting this panel together. It's great to hear about everyone's experiences and what leads to totality of evidence. So I am able to speak to a recent product approval for a rare disease with excuse me, significant unmet medical need. Our approval was based on one adequate and well-controlled trial plus confirmatory evidence. And we were able to establish that evidence in a pivotal trial with confirmatory evidence, which included strong mechanistic support, well-established etiology of the disease, the mechanism of action of the product, and also biomarker data. So a lot of that played into the approval. But there are a couple other success factors that I think are worth mentioning. We did receive breakthrough therapy for the product because it was a rare disease and unmet medical need. And that was really helpful because breakthrough therapy allows for increased communication with the agency, and the, excuse me, the expedited programs and pathways at FDA are really important in this context for encouraging rare disease drug development and making sure that treatments make it to patients as efficiently as possible. And when you're dealing with the challenges, which I know we all know very well, of rare disease drug development, the small patient population, heterogeneity sometimes, it's really important that those clear lines of communication are there and that industry isn't heading off on the wrong path regarding the use of certain clinical trial design or statistical analysis plan or the particular biomarker that you're using. You can have a drug that works, but a bad study design and your trial fails, and nobody wants to be in that situation. So it's really important that people, as you're able to, use these expedited programs and pathways that exist. And we were able to take advantage of that increased communication throughout the development process, and there were instances where we did need additional guidance from the agency, and we were able to get that in a timely manner and address any needs that were brought up and in a way that we did not have to delay our trial or our timelines, and so that was really important in making sure that product made it to market as efficiently as possible. Another huge piece of this was the patient voice, and the particular disease area that this product is for had an extremely strong patient community. And in this instance, 2 patient groups came together to develop patient perspective data on the disease, the impacts, the symptoms, outcomes of importance to them. And they did it in a very rigorous way, and they submitted it directly to the agency. And the FDA was able to use that information and mentioned it to use it as a additional source of information in their benefit risk analysis. So that was really important, and making sure that you're engaging with the patient community and really partnering with patients throughout the drug development process, not at the end, but early in the process and throughout the process, and making sure you're understanding what outcomes are important to them. And kind of related to that, I mentioned the biomarker data, and FDA has said this time again in guidances, and when you have your biomarker data, it's important to be able to link that to what's important to the patients. It's not just a measurement. You need to show how that measurement is clinically meaningful and how it's impacting your patient population and making sure that impact actually matters to them. So that's another key piece here. So overall, I think there are, in summary, three things, and that's early communication, frequent communication with the agency, showing how a change in your biomarker impacts outcomes of interest to patients, and making sure that you know what those are when you're developing the drug, and you really cannot understate the importance of partnering with patients and working with the patient community. Patient-focused drug development and rare disease drug development go hand in hand, and I think that's the way that it should be. Making sure that you have that data from the patients. Patient groups are extremely savvy. I know several people in this room have been involved in a lot of those activities. but they can really gather the data in a way that can support regulatory decision-making, and that's a really cool thing to see when it happens. So partnering with the patient community and making sure that they're partners throughout the process.
Dawn, just to add to that, you've got some examples of using endpoints and different special populations, and I wonder if you could elaborate on that, given the patient communities.
Yeah, I want to speak a little bit to feel, function and survive, and how you can use the same measure, but your responder definitions can potentially look very different depending on the pediatric population. So I think it's been clear already that we need to have such a clear understanding of the natural history, both naive and on standard of care. We have to understand the disease stage and the phenotypes very clearly related to what each of those phenotypes is going through. And so we're studying MPS diseases at REGENXBIO for gene therapy. And I wanted to share a little bit about how a responder definition, even within a specific disease and a diagnosis, you sometimes need to stratify in order to answer that question. And so we have neuronopathic patients that we obviously want to treat as early as possible because it's a neurodegenerative disease that's progressive. But at the same time, when those children enter into the trial, they have minimal disease symptoms. And so a responder definition for that group of kids may be that they're maintaining their developmental function within a range over time because we've caught them early, right? And then similar children in the trial who have more severe baseline disease presentation could look very different. And we're assuming that we're not going to have the same level of change in both those groups. And we're going to be looking at very different and maybe smaller changes in the group that has more severe disease presentation at baseline. So if I take those same outcome measures and I look at them in a different pediatric disease and say, how does the responder definition change? So when I was working at Alexion on hypophosphatasia, Strensiq was the drug. And it was the exact opposite there. The most severe kids in that indication were the very young ones. And they had, minimal bony integrity and muscle strength to move. They were ventilatory dependent, and feel function and survive was very different for that group. Those kids, the responder definition was survival. It was very different, and any skills they acquired was a bonus because of this incredibly severe disease presentation they had at baseline. Whereas the flip side, there was a attenuated or late onset version of hypophosphatasia in which we had to have a completely different approach in the outcomes. And in the younger group, it was easy for us to understand and the regulatory bodies to understand that survival and not having a control group made perfect sense for that group because it was obvious that they looked different than the natural history. But for these older kids, they came back to us and they said, no, this doesn't apply here. We need more evidence to understand how this group of patients looks different than the natural history. And I think in following that feedback, we were able to, fortunately, we had video recorded all of our assessments on ambulatory function. Good backup, right? That was a very good thing for us. And the PI that worked in that trial had been doing longitudinal video collection of ambulatory function on his kids for years. So all of a sudden, we had an opportunity to do central data analysis of these videos and meet this requirement to have some level of comparability between these less severe patients and the patients that were untreated. And so I guess my point here is that the regulatory feedback is incredibly important, but you also have to have such a sound and detailed understanding of the disease stage and the phenotypes, because one responder definition is not going to work across these groups. And it's independent of where you position your endpoints, right? It doesn't matter if you're going for accelerated approval or you're using one of these functional measures as your primary endpoint. You still have to have a high level of evidence and confidence in either one that you're showing meaningful, clinically important change, especially to the caregivers and the families. And so I think that's the lessons learned, is dig into those early stages, really use the FDA guidance and the roadmap to really understand, and people tend to jump to this end of the continuum and say, what outcome can I use? And I think you got to go back to your basics and just understand every nuance of how those kids function in the different groups and how that changes so subtly, whether they're, you know, in the delay stage, the plateau, or the decline stage. And so that's my
I'll
thought.
open up to others. I mean, endpoints are commonly a challenge in rare diseases, especially if there's been no regulatory precedence for their acceptance. Does anyone else on the panel want to talk about their positive and benefit experience around specific endpoints? I had to laugh, and Michelle were looking at each other when we talked about the roadmap.
Roadmap, yeah. I think everybody at Regenix does not want to hear me talk about the roadmap anymore.
I want to make a plug for a program that FDA has recently started in relation to the endpoints. We now have a new pilot program. It's called Rare Disease Endpoint Advancement Program. And we started it as a pilot following reauthorization of the Prescription Drug User Fee Act last fall. The program is up and running. We put information about it on our website in painstaking details. It explains what information is needed, what aspects of endpoint development would be important, and I invite everybody to visit that website, and if you put this Rare Disease Endpoint Advancement Program, you will see it there. We invite to send us proposals, and we receive a proposal per quarter, initially as a pilot. So that's the plug. Some assistance from the regulators on development of endpoints.
Yeah, just one comment, which is, I think a lot of times, we were just talking about this, I think a lot of times we focus on what are the right endpoints for our trials, and will FDA accept this endpoint? And I was saying this in the context of our AMP-ALS conversations. We've got to take it way back. We need a few steps. What are the measures, fluid biomarkers, clinical outcome measures, of how, you know, that are relevant in the disease, kind of carrying on from what Don was saying, at different stages of disease? And can we refine those measures? Can we identify new measures? And then we can figure out how to use them. Because I would argue that while neurofilament has obviously played a really important role in the approval of tofersen in the context of the surrogacy story, equally important is the role that it played in helping us control for heterogeneity. I mentioned how we got our enrichment strategy all wrong, and you can actually be more inclusive in clinical trials if you have a measure that is truly prognostic. And so I think that when we're talking about endpoints, sometimes we lose our way a little bit when it's really, let's bring the most objective and sensitive measures forward in different contexts of use.
Can I just add one point to hers? I think the next biggest challenge is we want to work towards shortening the time in development process and using accelerated approvals when we have the opportunity. And some of these biomarkers have tremendous opportunity to work across the lifespan, and we can see change across all of our patients, which is great. But then we also have to show that it's reasonably likely to predict clinical benefit. And that's where it gets very tricky based on what I said, because the magnitude of change for that young child that's in a normal range at baseline is going to be completely different than the magnitude of change that is going to be present for the child who's very severe at baseline. And that limits your ability to show this beautiful, strong statistical correlation, because it's going to be very different depending on those two groups. So I like the idea of having this one measure that you can use across your population, but then you still have to very much understand what are you going to do with that to show what is reasonably likely to predict clinical benefit and how it looks different by groups.
So we'll switch gears a little bit to Jesse. You've got an exciting gene therapy. So I don't know if you want to talk about that experience.
Yeah, so as I mentioned in my introduction, I spent some time at Bluebird Bio. I was there for 5 years, and during that time I worked on a product for the treatment of cerebral adrenoleukodystrophy, which is now known as Skysona. I want to be clear that I was not at Bluebird Bio at the time of the submission of the BLA, nor the approval. However, I did spend 5 years of my life working on the regulatory strategy leading up to the BLA. So Skysona is an LVV-based gene therapy, and it received accelerated approval in September of 2022. So the accelerated approval was based on an intermediate clinical endpoint, not a surrogate endpoint. And since I was there for the 5 years before, I can tell you that we never contemplated accelerated approval for this program. We had agreed with the agency on a clinically meaningful endpoint of major functional disabilities, which was the top 5, I think it turned out being 6 at the time of approval, deficits that these patients may experience. They were all drawn from the neurologic function scale. So our clinical development program and our primary endpoint was against natural history. So the clinical development program included 2 natural history studies. There was one natural history study, which was a retrospective natural history study, and the data that was collected there was not contemporaneous with the conduct of the registrational studies. And these patients were untreated patients. And that group of patients was intended to perform a benchmark against which we would compare the primary efficacy endpoint of major functional disabilities. There was also a second natural history study that was performed. This was a prospective natural history study, and it included patients who received allogeneic stem cell transplant for their CALD, which is the standard of care of treatment for patients who do have a match. And this study was run contemporaneous with the registrational studies, and we used the same endpoints, the same sites, et cetera. In addition to these natural history studies, there were 2 registrational studies, which included in excess of 60 patients with cerebral adrenal leukodystrophy. And per FDA guidance, all patients treated with Skysona were followed for a total of 15 years following their initial treatment. So there was also a long-term follow-up study. So in this example, FDA granted accelerated approval when this was not initially envisioned. So during the course of the BLA review, FDA had concerns about the ability to interpret the primary efficacy endpoint. And one of those concerns related to the comparability of the natural history population compared to the treated population. And specifically that the natural history population was more severe in their disease than the treated population. And so as I mentioned, this was data collected prior to the conduct of the registrational study. Treatment methods had changed with time, and we didn't have the same diagnostic methods, patients were not being screened at birth, for example, and we didn't have the same genetic screening methodologies. Given that, FDA requested that subpopulation analyses be performed. Those results bore out the overall conclusion that there was a benefit on major functional disability, but in a much smaller population of patients. In fact, over 80% of the Skysona-treated patients were excluded from the subpopulation analyses and aren't included in the Skysona label. So I think this case study illustrates some of the challenges in rare disease development that many of you guys have touched on today. Sometimes there is the need to leverage historical or prospectively collected natural history study. Sometimes it's not even possible to collect that data. And even if you do collect that data, which Bluebird Bio did, it may not be sufficient in terms of being able to well match patients, just given the size. So I think there's a lot of power in what CPATH is doing in aggregating data, right? So if it's just a company conducting a few natural history studies, it certainly doesn't have the same numerical power as many companies putting things together, or a single database that has a lot of data. I think in this case, or in all these cases, I think regulatory flexibility and allowing analytical methods to adjust for natural history and treatment group differences for rare diseases could be really helpful, especially when the landscape is constantly changing. And the first accelerated approval for a gene therapy actually just occurred in June this year with Sept's DMD program, which is very exciting for us in gene therapy. And we hope that the agency will be more open to the use of a surrogate versus an intermediate endpoint. And there's a lot of conversation today too about interfacing with the agency early to discuss these things. And I think there's often hesitance to have a discussion about whether or not something's can be a surrogate endpoint when there's no clinical data to support that the idea that there will be a clinical benefit based on the endpoint. But I do think that gene therapies are a space where it makes a lot of sense because the causal pathway of disease is clearly known. I think that their utilization of biomarkers as surrogate endpoints is definitely a reasonable pathway here. But for each rare disease, it's generally a novel surrogate endpoint, which adds challenges, unlike in oncology where, there are established surrogate endpoints that have been validated. And with time, I'm hopeful that the agency will also start to consider things like non-clinical data as supporting the idea that something could be a surrogate endpoint. But time will tell with that. So. They specifically said in the Sept basis for approval that non-clinical data did not establish something as a clinical correlate. So anyway, that's my thoughts on SKYSONA.
Larissa, do you have any comments on?
Sure. First of all, I would like to make a correction. Apologies for
no problem.
the first approval, accelerated approval of gene therapy happened in 2020.
You're right. It was a CAR-T.
Exactly,
Apologies.
and that was no problem. And
Yeah.
that was chimeric antigen receptor T cell therapy for treatment of mantle cell lymphoma. But as I was listening to the case studies and stories of different development programs, NDAs and BLAs, I was actually thinking of a proverb and the consequential aspects of that. And the proverb is all happy families look alike, but every unhappy family is unhappy in their own way. And so we're hearing today about happy stories, about successful product development programs. And they all have common features. And I think it is important as a takeaway message today is to understand and recognize all of these common features, because in preparation for future development programs, all of them are important. And I think each speaker actually talked about various aspects of their product development programs that are in fact applicable and would be counted in that list of common features for a successful development program. And that includes very good understanding of the natural history of the disease, genotypes, phenotypes, heterogeneity of the disease, the different types of progression in different subtypes of the condition that you're looking at. Very good understanding of what it is that one is intending to treat, and a great understanding of how a potential biomarker or a potential surrogate endpoint can actually fit in that natural history of the disease and what it can measure and how it can measure it. And one part that I haven't heard today, and that's still in the surrogate endpoint biomarker development piece of the success, is a really good validated assay for how the biomarker is measured. Because we see oftentimes biomarkers that make sense for product development, make sense for the natural history of the disease, even may be acceptable in some clinical practice measurements. But if you don't have a validated assay of what it is that is being measured, particularly if that biomarker is then eventually going to be used as an endpoint, then there may be an issue there. So measuring biomarkers in the way where you can have a reliable measure, and best if it's validated. Engagement of patient partners, that's also one of the crucial features of a successful development program. Understanding what it is that needs to be put into the package for substantial evidence of effectiveness. Obviously, for rare diseases, the agency has
said many times that instead of 2 adequate and well-controlled trials, we're looking at 1, recognizing the rarity of the condition, looking at supportive evidence, looking at how the study was conducted, looking at study design, video assessments, very well thought through instruments of how different, be them, clinical endpoints or patient-reported outcomes, how they're going to be assessed, right? So good instruments and study conduct where data integrity is preserved. So that's another feature which is important. And specifically related to gene therapies, with cell therapies too, biological products, many of them, there is a term that someone coined, and that's a living drug, right? In many cases for gene therapies, it's a single administration. For cell therapies, it may be repeat administration, but even with the single administration, we do expect longer effects of that treatment to be there. So a longer time of observation may often be needed. And sometimes you can't really make it, be it with surrogate endpoint or with an intermediate clinical endpoint, by the time when the product is approved and on the market, you would still want to see what are the long-term effects of that therapy. People talk a lot about curative therapies. When do we establish cure? We have to really look at absence of the relapse, at change in disease progression to the degree where there is no progression anymore. And the principle of faster, cheaper, better works best with semiconductors. But when we're working with biological systems, you really want to make sure that the effects of the therapy, the intended and the anticipated effects of the therapy are there. We had a meeting with stakeholders on April 27 of this year, and we talked about various methods for how one would evaluate both efficacy and safety in the post-approval stage, particularly when we have many products that are in development and people do not want to wait until the product is available to them. So we're looking at shorter times for development programs. Again, even if it's not with a surrogate endpoint, if it is with an intermediate endpoint, you still need to see the longer term efficacy. And we've received a lot of good feedback from those who participated in the workshop. We're planning on developing a guidance, on the topic. But one interesting part that I, took from that, we have certain time frames for how different product categories would need to be followed over time for, say, lentiviral vectors, 15 years, right, you mentioned. For some other vector-based products, it could be shorter period. But what we've heard from stakeholders is that they are concerned not just with understanding of the efficacy and safety, but also with life events, with life milestones. For example, effect on fertility. If somebody receives their gene therapy very early in life, in the first few years of life, even the 15 years that are there are not going to cover the period when they are of a childbearing age. And so with development of technologies, we all have to think about how we, follow people and better understand the effects of various long-term products that we give to people and how they affect their life. I guess that's
good. Jesse, did you want to comment about post-marketing requirements for gene therapies in relation to the discussions that you've had?
Sure. So for Skysona, which I just spoke about, there are 2 studies that need to be performed for the accelerated approval. And one of those studies is to enroll 24 newly treated patients who have more advanced disease. which may be a very big challenge considering that there is newborn screening efforts, there's genetic screening. The likelihood that a patient today will be treated later in the course of disease is much lower than it was previously. So I think, sometimes there are challenges associated with the requirements that are put in place for sponsors following approval. In addition to that study, there is a post-marketing requirement as well to conduct a prospective observational study in 120 patients, which needs to be completed in 2047 to assess long-term safety risk as well as the risk of secondary malignancy, which did appear with Skysona. That as well is, I think, a big challenge and ask, considering it's anticipated that approximately 10 patients per year will be treated with Skysona. So, that's 12 years of patients for enrollments. And I totally agree with everything you're saying, by the way. I'm not, but I do think that we need to, because sometimes I think accelerated approvals occur later in development versus, discussing upfront and early, like that you're headed towards an accelerated approval. I think sometimes there isn't time for conversation about what would be a meaningful study and time to allow the sponsor to sort of like undertake and think through that more. But at any rate.
Yeah, I guess I just wanted to maybe discuss a couple of items here. One is dealing with heterogeneity of the disease, right? So that's another kind of, continuing on the same topic, common feature of successful development. So you have a very heterogeneous disease. There is an effect in one subpopulation, and maybe trend in another subpopulation. You really would like to see what that effect looks in the subpopulation for which you don't have sufficient data. And so here we have this ability to monitor the other subsets of people, the subgroup with pyramidal tract, right, disease, and the other subpopulation. So there are ways of how one could look at one heterogeneous disease, maybe see initial efficacy demonstrated in one patient subgroup, and follow other subgroups over time to see what happens with the treatment, in those people. So that's one. And for the safety requirement for the post-marketing study, I'm sure you know well, Skysona did have a signal. It did have a signal with myelodysplasia observed, and so we owe answers to people who are being treated or who are potentially considering treatment with novel therapies. And when we don't have enough data about safety signals that are seen in pre-marketing, we try to follow those patients and try to follow those who are treated with the product to see what is the actual incidence and frequency of potentially very devastating side effects, because obviously with gene therapies, with lentiviral vectors, we are worried about secondary malignancies. If it takes a certain number of people or a certain period of time to understand if the risk of second cancer is there, I think many people have already answered this question as, yes, we do need to have follow-up.
Mark?
I was just to say, around the whole accelerated approval strategy, before I was working on tofersen, every oncology asset I worked on, accelerated approval was our only strategy, primarily, right? Because you're targeting a late line indication, but there's just so many differences that I think are important to point out before we get to the similarities, because virtually all oncology accelerated approvals are based on some form of response rate, right? Durable response rates in some form, and that is exactly what you're looking for in the same data as for your go, no-go decisions or your expansion of phase 1 to have something that's potentially registrational. So when you're going into your first of human study, you're hoping for some partial responses, right, in the heavily pretreated patients, or if you get a complete response, then you start to expedite further. So it becomes part of your primary strategy. I think some of the complexity that we're talking about here when it comes to a biomarker as a surrogate endpoint that's reasonably likely to predict clinical benefit, often we are having the same discussions with tofersen as we were beginning to determine what was substantial evidence of effectiveness was a biomarker that's never been used for regulatory decision-making before, right? So there's some clear differences there in the data and the evidence you need to build throughout development. Ultimately, I think it comes down to the same criteria when you try to understand what is reasonably likely to predict clinical benefit. You have your biological plausibility and your empirical evidence. I completely agree with what you were saying about the need for robust assay development, particularly as the effect on a biomarker becomes less. Then, of course, you need an even more kind of robust assay. So I think it's fascinating. I think it's the most challenging is for when you're going first. I worked on assets in small cell lung cancer where we had overall response rates that is used commonly, but in that particular indication, when you have a tumor that's doubling in size every few months, it's not reasonably likely to predict clinical benefit. So there's so many learnings, I think, in the successes and also the products that don't make it. But it's complex, I think, and when you're talking about a biomarker to be used in regulatory decision making, you often don't know until you get there, until you know what your empirical evidence is, and you're still working through the biological plausibility. And that was part of our advisory committee meeting strategy, was to try to kind of weave that tapestry together to say what is reasonably likely and how are we demonstrating it.
Any other comments?
I'll just add, that was a great discussion, and I think, to quote Michelle Campbell, but engage early and often. And when we're talking about endpoints, whether it's biomarkers and surrogate endpoints or choosing the right outcome measure for a rare heterogeneous disease, the earlier we start that conversation, the better we can kind of help you have a successful study. I think when we talk about assay validation for biomarkers and surrogate endpoints, the earlier you plan to use a biomarker or to rely on an intermediate clinical endpoint for a trial, the better it is, because you can plan for that accelerated approval, and then the post-marketing requirements and the things are easier to successfully complete. I think we heard two stories today of kind of last-minute accelerated approvals, but they're most successful when you know that early going into it. And then sometimes that study to confirm clinical benefit can be almost done, almost completed by the time you reach accelerated approval, and you don't have to worry as much about how do you complete that trial in the face of an approved product and dropouts and enrollment and recruitment. So I think if you can engage early, the better you'll be. And I just wanted to clarify one point that comes up a lot in just terminology is confirmatory evidence versus confirming clinical benefit after an accelerated approval. And they're two very different topics.
I just wanted to take us back to the patient piece for a minute. We saw some great examples of how you incorporate patient/caregiver input into your drug development pathway. And I think industry as a whole tends to be really good at capturing the patient diagnostic journey, the patient journey. I think that's important, but I think that's only a tiny piece of what we should be doing with caregivers. Meaning that I think we're totally underestimating what they can do, how involved they can be, and we should be asking them the really hard questions. They know the diseases better than any of us, and if you come to them and say, what is the relevance of these items? How do these items, how meaningful are they to you in your world with your child? They will give you incredible examples of the item relevance and how that impacts them in their daily life with their children. And that's part of your outcomes. That's part of your narrative and your story. And the other example I think that should be highlighted is PPMD and their patient preference work. And so in gene therapy, the work that they did on a large sample of caregivers related to the level of risk that they're comfortable in, how that differs by phenotype and disease progression. It was so informative and so important, and that's a huge example of how everybody in the DMD space can benefit from understanding how caregivers make decisions related to enrollment in a study. And so I'd like to see us all kind of shift away from going to vendors and seeing these beautiful PowerPoints on the diagnostic journey. That's great, but that's not the real questions that we should be asking of these caregivers. We should be digging in and really using them as a resource that's powerful and meaningful and can really change the direction of our development pathway.
And to add to that, I don't think it should really stop there. You know, when you're looking at post-marketing requirements and follow-up studies, you know, you still need to engage with the patient population to reduce patient burden and help make sure that you're actually you're designing that follow-up in a way that you can get that information from patients and really help retention. So it doesn't just stop with approval.
Right.
Sorry, just back to the SKYSONA for a second. I didn't mean to uncover two contentious sides of the sponsor FDA claim. I do, just in terms of patients, want to say I do think that FDA exercised an incredible amount of regulatory flexibility in finding a path forward for SKYSONA so that patients and caregivers can make an informed decision about whether or not this is the right product for their patients, but it is available. And I do think FDA was incredibly creative, and it certainly is not the perfectly executed program. There absolutely was a MDS that appeared in a risk of malignancy, and gene therapy should not be taken lightly, right? It's a one-time treatment. You can't get something again, So at any rate, I think FDA did do a really amazing job in terms of the regulatory flexibility that they exercised there.
Yes, I'm jumping backwards, but this has been on my mind quite a bit lately, and it's probably an obvious statement, but I think as we're thinking about new endpoints, and using ALS as an example, but this isn't unique to ALS, so much of it comes down to what you're anticipating with the therapeutic landscape. And as we look at ALS, when you look at not Biogen's work, but the whole community's work of therapies that are in development, and as we understand the disease better, as we're developing more targeted therapies, we've always sort of operated in a world where what we were hoping for was slowing of decline. But if we identify and develop therapies that can actually lead to recovery of previously lost function and strength, the existing gold standard endpoints are not going to be sensitive to assessing what, I'll coin improvement across those measures. And there are some really interesting examples. We had physical therapists that were looking at tofersen-treated individuals and seeing things that we were not seeing with the endpoints we were measuring. And all of a sudden, it made me think, these are endpoints like the 10-meter walk test, the TUG, the Berg balance test that are being used in many of the diseases that we're talking about today that we had never considered in ALS. And maybe that is something that we could learn from each other and really try to talk more cross-TA when we're considering new endpoints in the future.
I guess I'll open up for any questions. We're using up all our time, so I want to make sure that if people want to give a chance to discuss some things, please up to the mic.
My question is about for rare disease. So we might have some clinical trials that are global in EU and in the US. So in having this dialogue with the FDA, so should we have a dialogue with the FDA separated from the EMA? Or is there a kind of coordination if one, for example, refuses to have a liquid biomarker as endpoints and the other authority is okay with it? So how does it work? Is there a coordination? Should we talk to both separated? So that's my question.
So I'll try to address this. Obviously, because we're in different regions, each country or different countries, EU, includes a number of countries. They have their own laws, and regulations are extensions of laws, so the regulations are different, obviously. In many cases, particularly for rare disease, we try to collaborate and converge on the requirements for product development, but each country is still under their own regulatory framework. However, because many studies are multinational studies, they're multi-regional studies, oftentimes what we see is that the same one study can be sent in to both FDA and EMA, and for example, right? Maybe to other regulatory authorities too, but I'm just talking about FDA and EMA. Sponsors typically have separate meetings with FDA and EMA under processes for each of the regulatory agencies. We do have a process that's called parallel scientific advice with EMA when we can provide parallel advice, yet it is still not the same advice. It could be converging, but wouldn't necessarily be the same. In some cases, it could be very similar, if not the same, but it's not really the same advice. It's just to make sure that the requirements for each region have been met and not at the expense of, doing something very large or not feasible. So we try to work together with other regulators. We also have various bilateral meetings where without sponsors we can discuss development programs for different products and just exchange opinions on what could be done or what has been done and how we assess different development programs. We have clusters in different areas in patient advocacy and rare diseases in various therapeutic areas. So we do collaborate to an extent, but again, every region is under their own regulations.
Hi, my name is Yin YIN from Belgium. I'll say hello to you guys after the meeting. I have a question about for the FDA, for the questions you post for the advisory meeting. I do not watch all the advisory meetings, but I did watch the Toferson because it's about, and I think the first question was about the accelerate approval, and it was very clear, and you got unanimous votes for that. But there was a second question, and the question was asking the advisors whether the evidence was significant. And then a lot of advisors kind of were stuck by this significance. And then the question changed from significance to convincing. So they still had the same trouble with the adjective. So I'm a statistician. I think I try really hard when I work with teams, try to avoid using adjectives, use numbers. And because, because when people say, oh, it takes too long, how long? Years or days? Or when they say expensive, how much money you're talking about? Sample size, or too many people or too few people? How many are we talking about? People have different assumptions. So I think the question was kind of using adjective, just really confused a lot of people. Even for English major, probably they have trouble to understand what the difference between significance and non-significance. And I think FDA Clean Farm presentation did a good job, and they have this good story and incorporate all the numbers to have this package. And then I look at it, two people voted for yes. And their reason was if you even approved by this accelerated approval, the patients can still not afford the drug because if not the full approval, the insurance is not going to cover it. So it's really kind of, so those two advisors really kind of stand from the patients kind of in their shoes and to think about it. Other advisors were thinking about what convincing means. So I think that the question should be maybe like, are you going to prescribe this drug for your patients? I think that probably, that's their job. It's not to interpret significance or, adjectives.
I appreciate the comments, and I think we learned a lot from that advisory committee as well. I think there was the attempt, the application and the discussion was about accelerated approval, and that's how the NDA was submitted and reviewed. And there was a thought that, did anyone on the advisory committee feel that the data was substantially convincing that it should get full approval? And that was may or may not be poorly phrased, but it was an attempt to see if the data was there according to the committee. And so that was how we phrased that question.
Tim?
Yeah, thanks, Collin. Tim Franzen from CPAP and Faegre Drinker. Thank you all for some really instructive comments about best practices. And that's always important in learning. Something that we may benefit from as well as the converse. So experiences you would have had that show the need for regulatory inflexibility that are unreasonable, and the opportunities to learn from those. So would the panelists care to comment on that?
We have to bear in mind who we have in the room and on our panel as well, I think. But I mean, it's a good question. I think that this came up a little bit when we were discussing the accelerated approval reform, right? A little bit about, are we taking enough risk with the regulatory pathways, right? And then you can inherit from there your question, really. I think we did a lot of work as a company and as an industry group to assess successes of the accelerated approval pathway. So programs that were approved as an accelerated approval, then went on to confirm benefit. Then we looked at products which did not confirm benefit, and another regulatory decision was either made to withdraw or in some circumstances leave them on there. And I think looking at it from that lens, you come up with, if you will, about a failure rate of about 8 to 10%. So you can see for all accelerated approvals from 1992 onwards, right, the failure rate is about 8 to 10%, i.e. programs that gained an accelerated approval then didn't confirm benefit. To me, that's quite a good metric to look at. I mean, if that number was 2%, right, then I would say maybe we're not taking enough risk with the pathway. Then that would suggest to me that maybe there's too much inflexibility, right, around what is reasonably likely to predict clinical benefit. Conversely, if 40% of them did not go on to confirm benefit, then maybe we're being a bit too liberal with how we're defining reasonably likely. So just from that metric, to me, 8, 10, 12% feels about right, holistically across all development programs. So I think it's case by case. That's accelerated approvals are typically on the more aggressive end of regulatory strategies, and I think that's a good way to look at success and failures, to look at that kind of generational impact of that piece of legislation that enables that. I often hear folks asking for more consistency in regulatory decision making. I hear that mainly from industry. It's not a point of view I subscribe to. For all the examples we just discussed today, there's so much nuance in there, right? There's so much that the FDA has to review that is completely new data, completely new context of use, often under a priority review because these are the more serious, life-threatening conditions of highest unmet medical need. So I think it's a difficult balance. So I think you have to take a step back and look overall. Metrics will tell you one thing, right? But in terms of enabling FDA to make decisions, right, that's on industry as well, right? To be, as we were discussing, how is one defining substantial evidence of effectiveness? And if we had not been successful with the tofersen application, I would look at that as a failure on our part, that we haven't enabled the agencies to make the decisions they're doing. So it's a difficult question to answer. I'd look at accelerated approval metrics, but maybe others have other thoughts.
If you wanted to say that.
Yeah, and I'm not just saying this because Dr. Freilich is sitting next to me, but I always say to the team, the agency knows more than we know, right, about more products. And I love the example that when we were designing the phase 3, we were talking with the division about the sample size and requiring a two-sided alpha of 0.05, and I said, I don't think we can enroll 100 people. And I remember Dr. Dunn saying, you need to try, and then we can have a conversation. And in hindsight, had we not tried, had we not enrolled 100 people, I don't know that we would have been able to interpret the data set. So it's one of those good examples. I wasn't excited about the feedback at the time, but I think, again, trusting sort of the process and keeping all of us, keeping each other in check, I think is really important.
Emily, did you want to say something?
Yeah, I was just going to say, we try to be flexible when we can, but we still need to meet that substantial evidence of effectiveness. And I think we don't pull it out of the air. So, when you talk about inflexibility, we are going to stick to the data that's needed to show that a drug is both safe and effective.
I think we need to be cautious in interpreting successes and failures in previous filings in terms of how they apply to us, because I think it's a new world. Rare disease is different now. Like, I've been doing rare for 20 years. The knowledge people have, the approach the FDA speaks to is very different. And I also think we're doing gene therapy, and that's a brand new world. And so I think we have to believe that we're all learning together, and it's a work in progress, and we just need to take it one step at a time and do your homework, right? Like that's my takeaway. Like there's no golden ticket or easy pass that you're going to get. There's tons of guidance documents out there that will guide you. And yes, we can't meet all of the boxes in those documents, but that's an okay place to be. That's an okay place to say, you're asking for this, and this is why this doesn't work in my disease or in my subgroup or in this model. And this is what will work. And so I think learning together and using those guidances, and our job is really hard in Rare. This isn't a walk in the park, but so is theirs. And I think we just need to do this journey together and take it one step at a time.
Melissa, I'll give you the last comment.
Yes, I just wanted to say in relation to the accelerated approvals metrics, the accelerated approval pathway has been there in the regs since 1992. We haven't really used it as much as we're using it now, and I think everybody recognizes that with the change in the landscape of product development and with more rare disease product programs entering the regulatory field, we're seeing an increasing number of accelerated approvals. And I think one important piece to recognize is that once the accelerated approval happened, the game is not over, right? We have to see the verified and described clinical benefit. And one part of some inflexibility that may have been observed in the past was that when there is no demonstrated and verified clinical benefit, it sometimes is difficult to withdraw the product from the market. And so I think, moving forward, as we grant more accelerated approvals, there is a need for general recognition of all stakeholders, of industry and patients and policymakers, that once something isn't shown or the substantial evidence of effectiveness based on clinical outcomes is not there, then the next step is not go into a prolonged process with FDA in discussing, how we can remain on the market. The next step is to, withdraw and maybe repurpose or redevelop or do other things. So I guess, we will see how this issue would unfold, again, because we're granting accelerated approvals. But the lifetime of the product is important to consider here when we're discussing these FAST programs.
Okay. I want to thank the panelists for all their time and lending their expertise to this group, and I'll pass it on to them.
Great. Yes, what a wonderful panel. Thank you, everyone. Okay, so we'll be heading out to lunch. We have lunch set up right here out in the hallway. Please go help yourselves. We have about an hour, a little bit under an hour for lunch. You are welcome to bring that back in here and eat. There's plenty of spaces out in the general area, but this is a free time for lunch. We will be back here around 1:15 to start the afternoon sessions. Thank you.